Good morning. I think we'll make a start. If I can ask you firstly all to mute your microphones and turn off your camera. We are planning on recording this session, so it just maintains a bit more privacy for you and obviously eliminates background noise. So thank you ever so much for joining today. We've got a wonderful lineup of people today talking about rare diseases, and I'm going to show you who we've got speaking. So firstly, we've got Professor Tim Barrett, and I'll introduce him in a second. And then, as you can see, we've got quite a packed schedule, and we'll introduce them all separately. If you do have any questions, you are welcome to put them into the chat, and we will try and answer them at the end, or we might try and answer them within the chat function. Tim, you are here. I've just had a quick look at the list, and I think I can see you.
Yeah, I'm here. Yeah, it's all good.
Wonderful. Yeah. Great. Okay. So first things first, I'd like to introduce Professor Tim Barrett. So Tim is a professor at the University of Birmingham, where he teaches various things about rare disease, and he's especially interested in rare forms of diabetes. And when he's not doing that, he's also a consultant paediatrician at Birmingham as well. So he's very busy, and I'm very, very grateful. Tim, thank you so much for joining us today. Would you like me to do your slides for you, or would you prefer?
I'll share my screen if that's okay. If that's
yeah,
all right.
No problem. Thanks, Tim.
Let me just get the technology going. Check I can do this. Let's have a look. Here we go. Can you see my screen now?
Yeah, I can.
Great. So, Vicky, thank you so much for inviting me to present on this. This is a real privilege to do so on Rare Disease Day as well. And also thank you for putting up with my lectures, because you've been on the course with me recently, and you asked very clever questions as well, which was great. So thank you that side. So what I was going to do is, just as a brief introduction around rare diseases, just to give some background from my perspective as a children's doctor. And when I think of rare diseases, I think of things that are relatively common for us in pediatrics. So cystic fibrosis might be an example of one. Probably affects about 1 in 2,300 children. So that's one end of the spectrum for a rare disease, anything that affects less than 1 in 2,000. The other end of the spectrum is a lad I've looked after, I've changed his name, called Kevin, with Alström syndrome, that affects less than 1 in a million. And indeed, there are rare diseases even rarer than that. And you put those together, there's about 8,000, or perhaps slightly more than that now, rare diseases, anything ranging from 1 in 2,000 to less than 1 in a million. But collectively, that represents 6 out of every 100 people. So that's a lot of the population, and a lot of burden of illness, and a lot of cost to the National Health Service. So a really important issue for us here. And then I'm thinking about Central and South geography for the Genome Medicine Service here. We cover the West Midlands, Oxford region, Thames Valley, down to Wessex and the South Coast. And I think the population is estimated about 18 million people. So if you think of how many affected in rare diseases, that's something like over 1 million people with rare diseases in the Central and South region. So that's a phenomenal number that we have to address. 80% of rare diseases are genetic, usually monogenic as well, and about 20% no known genes at the moment. 80% of them, in fact, probably nearer 95%, have got no dedicated treatment or a cure. So there's a massive lot of work to do to try and find treatments for these. And 30% of rare diseases affect children, which is why I got involved with this area, of whom about half will die before 5 years of age, because we don't have adequate treatments for those at the moment. So that just sort of gives a scale of the issue for our geography that we have here. When I started working with rare diseases, this is one of the first families I met. Charlotte and Matthew, they were 2 children with Alström syndrome, in fact, teenagers. And I'm going back to the millennium, really, about 2000. This poor family, they spent about 14 years trying to get a diagnosis for their children of Alström syndrome. And Rare Disease UK put out some really important papers regarding the patient perspective on rare diseases from about the early 2000s, saying that on average, families had to have about 7 visits to different doctors to try and get a diagnosis. The time from developing symptoms to an accurate diagnosis was up to 5 years sometimes. 40% of people with rare diseases described having their diagnosis missed or misdiagnosed. And of course, this has a massive toll on the emotional well-being of people, their carers, their families, and of course, delays to treatment or supportive treatment to look after people. So a massive impact on quality of life. That's all been summarized as a diagnostic odyssey. And what I want to describe in the next 20 minutes or so is some of the advances that have reduced that diagnostic odyssey down. To do that, I'm going to take one example, rare diabetes, because it's an area that I work in particularly, and I'm going to give a couple of examples of how diagnostic odyssey have changed for that. But just to understand where that fits in, we all know what diabetes is. It's the sugar levels are too high in the blood. Type 1 diabetes is the commonest one affecting children and young people when your pancreatic beta cell stops making insulin and you have to treat it with insulin injections or a pump. Type 2 diabetes is the commonest one affecting adults, very common, where the pancreas is still working, but it can't make enough insulin to keep up with the demands on the body. Probably affects about 10% of adults. There's a small amount of genetic influence to it, but there's a lot of environmental factors. And the bit I'm going to talk about now is monogenic diabetes, rare, purely genetic forms due to genetic issues within the function of the pancreatic beta cell. So my example here is a girl that I met. Again, I've changed her name when I was on call in the Children's Hospital in the late 1990s. She developed chickenpox, and sadly for her, had an unusual complication of aplastic anemia, where the bone marrow was not making red cells and became very transfusion dependent. And while she was in the Children's Hospital in Birmingham, she developed sugar in her urine, and that's why I was involved to go and see her. And she ended up having diabetes that needed insulin treatment. When taking a history from her and her parents, it was apparent that she'd got profound deafness from pretty much from birth, and consequently speech delay as well. Now, this is a recognizable collection of symptoms that has been described before in the literature of anemia, diabetes, and deafness. And we were able to give her a bespoke treatment for this vitamin B or thiamine vitamin, and that helped reduce her insulin requirement, her transfusion dependence. It was a clinical pattern called vitamin-dependent diabetes, which is very rare, very unusual, affects about 1.25 million children. It's recessively inherited, and there's about 30 or so families in the UK, all coming from the Indian or the Pakistani side of Kashmir. It's due to mutations in the thiamine transporter into the cell, and it's this classic triad. I'm mentioning it because we had to diagnose it through the symptoms, and because it had previously been described, we were able to put our families together with families from Israel and other areas and identify the gene, which was published in Nature Genetics in 1999. And having found the gene, we were able to use Sanger sequencing to sequence all the UK families and identify where, and the cartoon on the left, you can see it's a transmembrane folate carrier protein, where each of the mutations in the UK families had occurred. And on the right, you can see the old-fashioned Sanger sequencing that used to be done, looking at the different nucleotides, sequencing these little peaks here and finding where the mutations were, putting families together with those in Italy as well. That was a long-winded process to be able to find that gene and then do that Sanger sequencing at the time. What's happened since then, of course, and perhaps one of the best things that Prime Minister David Cameron achieved when he was Prime Minister was to fund the 100,000 Genomes Project. And this really revolutionized and took advantage of the advances in technology for next generation sequencing. It was announced in 2012, samples started to being collected in 2015, and overall it collected over 50,000 samples for rare diseases, mainly trios, so mum, dad, and affected child. But importantly, taking advantage of next generation sequencing, it could sequence the genomes of patients and develop panel apps, gene arrays or disease arrays that could look for targeted genes and mutations in individual conditions. And this is now being translated, as you know, into NHS care, thanks to this stimulation of this 100,000 Genome Project, which has been a great advance. In the case of rare diabetes, Sean Ellard, professor of genetics in the lab in Exeter, published a really good paper early on in 2013, showing how this virtual gene panel, using data from exome sequencing, genome sequencing, could replace the old Sanger sequencing that I showed by having a panel of all the known genes that could cause monogenic diabetes. And the gene that affected PREAT, this vitamin-dependent diabetes, is on there. It's SLC19A2, it's folate carrier. And through this mechanism, all these genes and variants could be tested at the same time and get to a diagnosis much, much quicker. So really, really great advance to do so. And what it's really done is shown how things have changed. So when we looked after PREAT in the before 2000, we had a defined clinical phenotype, i.e. early onset diabetes, development of anemia, in this case here. We had to do a genetic test to look for the gene based on the clinical features, and do Sanger sequencing to confirm the diagnosis. Since the 100,000 Genomes project and now in the next generation sequencing era, you can have a very poorly defined clinical pattern, as PREAT would have had, but you can test for all the known genes that will cause monogenic diabetes at the same time and get a genetic test result very quickly that can then determine what the treatment should be. So in this case, identifying that it was a folate carrier, you've then got a logical reason why to try Pharmin vitamin to be able to treat it. So the genetic result can now predict what clinical features will develop and what management is going to be needed in the future. So really, it's been such an exciting time to be involved in clinical medicine, to see this change in practice in the last 25 years. This has now gone to the next stage, really, and work done by Lucy Raymond in Cambridge and developed by my colleague, Dr. Swati Naik, here in Birmingham and other areas, has utilized this R14 panel for genomic testing in very ill children. And this is applied for children who are attending pediatric intensive care units and indeed neonatal intensive care units now, where they've got poorly defined symptoms of illness, whatever the cause, but it's producing a significant diagnostic yield for an underlying genetic causes. And it also determines what the best treatment is. So of the diagnoses identified through this genetic testing panel, 2/3 of them are affecting the treatment decisions and providing the best treatments for these available. So really, really exciting development that can be applied now to children with undiagnosed acute illnesses that can help find the diagnosis. That, of course, has then gone backwards to prenatal, before birth. And this is a slide shown, courtesy of Stephanie Allen, Dr. Stephanie Allen in the Genome Laboratory Hub here in the West Midlands, who's been involved in the R21 panel and prenatal exome sequencing. So in this case, it's sequencing the coding parts of the genome for genes that are known to be associated with human illness, and analyzing those that are clinically relevant for structural anomalies and some indeed now metabolic anomalies that it's important to diagnose before birth. So this fetal anomaly panel is now linked to these genes that are really important for a fetus and actually enable rapid analysis early on to support the care of pregnant women and affected fetuses. Going forward, the issue about prenatal diagnosis has always been having to use amniocentesis, i.e. sticking a needle effectively in through the uterus to the cord of the fetus and taking a sample that way, which of course is associated with risk of pregnancy loss by doing that. But because of great advances in technology and being able to identify the fetus's DNA going around in the maternal circulation, albeit in very small quantities, has allowed the opportunities for non-invasive prenatal diagnostic testing. And again, this is being developed further and very excitingly in the labs here in Birmingham by Stephanie's team and others. It can be used for to detect fetal sex, of course, but really importantly now, single gene disorders, and that's the development where it's coming in, so that if there's a family history of spinal muscular atrophy or Duchenne's, etc., non-invasive prenatal diagnosis can now be used on the NHS to try and understand these diagnoses very early on in pregnancy. It can be done. So I've talked really about some of the improvements in diagnosis around prenatal structural anomalies and then the critically ill children. The bit in between has been the NHS newborn screening programme, which, as you know, is really limited to about 9 or so conditions based on a heel prick blood test done by midwives in the first week of life. which has included more recently sickle cell disease and cystic fibrosis. But what's really exciting now that fills this gap between prenatal and postnatal is the Generation study. So really exciting initiative led by Genomics England and piloted across hospitals in our geography to actually screen newborn babies for a range of genetic conditions that can be treated early on in life. So with the theme of the Diagnostic Odyssey traditionally, and these slides very kindly supported by courtesy of Karen Creed, you'd have a healthy baby that unfortunately develops symptoms due to a rare disease, a genetic rare disease. That baby then has to be referred to hospital specialists who can then undertake tests, and eventually the baby will get a diagnosis. But all that time is then wasted really before treatment can start until the diagnosis is available. The Generation study aims to try and remove those three middle steps by screening all healthy babies and identifying those with a rare genetic condition before they develop the symptoms to get that diagnosis early enough to be able to actually do something about the condition. Now that's great, but there's 8,000 rare diseases and not all of them are amenable to current treatments or intervention. So there's a real issue to see whether this is going to be a useful option to roll out for diagnosis within the National Health Service. So the Generation study aims to evaluate the usefulness of screening newborns and how feasible it is for a number of childhood onset rare conditions using whole genome sequencing, trying to understand how this genomic data can be used for future research, developing new treatments and diagnostics for NHS patients, and understanding the potential risks and benefits of storing a baby's genome over their lifetime. And of course, this raises really interesting ethical issues about what a baby's genome might be used for after that person has grown up 40 or 50 years later with later onset diseases. I've had lots of discussions with colleagues in the rare disease community and patient support groups asking why isn't my condition part of that genome panel? And the four principles that they've really used to select the 200 conditions that are on it are, first of all, there has to be really clear evidence that the genetic variant or variants cause the condition and can be reliably detected. Secondly, a high proportion of individuals who've got this genetic variant would be expected to develop symptoms. You don't want to necessarily pick up people who've got a variant who will never go on to get the disease. Thirdly, they've particularly selected conditions where early intervention can make a difference to the outcome. So where there's a clear management pathway to look after these children should they be identified with a rare disease. And fourthly, it's been really important to make sure that conditions are selected where there can be equity of access for the interventions, i.e. Supporting all diverse communities that we have in our four nations, not just one particular segment of the population. So looking at the update nationally, the first site started recruiting in March, so almost a year ago, just over a year ago. There are about 25 sites live across England, and the aiming is to open about 30 or 40 trusts and to try and recruit something like 100,000 babies to this pilot study, and it's open until the end of next year. So where we are in Central and South, there are two sites open in West Midlands, a site open in Thames Valley, and Wessex is just about to open. So a number of sites. The recruitment in Central and South has been over 1,300 so far, and they've got almost 1,000 samples to date. So thank you, Karen, for sharing this slide at the moment. Nationally, over 5,500 families have given consent. 3,500 samples have been taken, mainly from cord blood, but some from heel prick as well. And so far, out of that number, there's been about 18 rare diseases suspected, which are waiting confirmation data, and then referral on to the relevant clinical teams who will be able to look after them. So a really, really exciting project, and it's going to be really interesting to see whether this is going to be shown to be feasible and useful enough to be able to roll out nationally in the next 10 years or so. The final part of this, I wanted to talk briefly about therapies. So I've talked about how the improvements in genomics has led to faster diagnosis and reduced the diagnostic odyssey. But 95% of rare diseases still don't have treatments. So I'm very briefly going to talk about three initiatives that are going on within our geography the LifeArc Centre for Rare Disease Trials, Nucleic Acid Therapies Accelerator in Oxford, and the Platform for Nucleic Acid Therapies, which involves colleagues in Southampton. So the Rare Disease Trials Acceleration Platform is a consortium of universities, including Birmingham, Newcastle, and Belfast, funded by the medical charity LifeArc, to try and address some of the challenges faced by people with rare diseases. Above the blue line, you can see some of the issues around getting a diagnosis, previously low diagnostic rates with variants of unknown significance, lack of information about disease pathways, difficult to recruit to patient trials, limited trial capacity, and then inequitable delivery, and then delays in regulatory approvals in the NHS. This LifeArc Rare Disease Translational Acceleration Programme aims to tackle the bits below the blue line on that right-hand side under the brackets. So it's developing a recruitment hub for patients to self-register with rare diseases, put in their symptoms, find out what clinical trials are available in their area, and with their consent, link their records to their NHS real-world data saved in a secure data environment in university hospitals in Birmingham. Secondly, to try and develop patient-relevant outcomes for clinical studies, things that matter to patients, not just what matter to doctors. And thirdly, to try and develop rare disease clinical trials capacity in the UK so that we've got dedicated teams with expertise in how to design a trial for a rare disease that needs as few patients as possible, that can use existing historical data, and that can try and reduce the need for a dummy treatment or a placebo arm where possible. And then the other aspects are speeding up trial delivery at sites across the UK and working with the regulators to try and get treatments adopted into the clinic faster than we've done so far. And there's a big patient and public involvement and engagement element, equity and diversity, and training for PhD students as well. Secondly, what a really exciting area of treatment are the RNA therapies being developed. And these are small bits of nucleic acids that are used to translate from DNA into proteins. And these can be either used to silence overactive transcription of deleterious proteins, or to boost the production of missing proteins that are needed for pathways. Some of these will actually work as ASO is an antisense oligonucleotide. And of course, the mRNA, messenger RNA that we're used to are those that were made for the COVID vaccines. And these are now being used to develop treatments for inborn errors of metabolism. In Oxford, the Nucleic Acids Therapies Accelerator is a fantastic investment by the UK government, led by Nick Lynch and previously by Matthew Wood, looking at these technologies to try and make them safe and to be able to deliver nucleic acid therapies and mRNA therapies to patients with common and rare diseases. So really exciting piece of work that's going on to look at these. And some of these have been adopted already by companies such as Moderna, who are looking at mRNA therapy for rare inborn errors of metabolism. And some of these are in clinical trial within our geography at the moment. And then secondly, the UpNat is a Medical Research Council funded platform for developing nucleic acid therapy for rare diseases. And this is a consortium between Oxford, London, and Southampton. And it's assessing rare genetic variants in families for suitability. And Professor Diana Barales in the Southampton region is heavily involved in this, validating some of the genetic variants and selecting those families and those variants that are going to be most amenable, and some of the earliest ones that can be used for antisense oligonucleotide therapy. So really exciting work going on within our geography for these new therapies. So in summary then, what I hope I've shown, and I think is really exciting, is how genomics is being integrated in all aspects of diagnosis and reducing that diagnostic odyssey, increasing the proportion of affected people who are now getting a diagnosis, thank goodness. And we now have some prospect of treating some conditions before they become symptomatic through the Generation study, and Central and South is leading the way in the pilot centres for rolling this study out. And what's really exciting, I think, is we're beginning to see on the hospital side some of these genomic therapies coming into clinic. We've had early examples for spinal muscular atrophy and diseases like that, but there are more coming along their way, thanks to the work going on in Oxford and Southampton to deliver it. So I hope that's given a bit of an introduction, an overview of where things are from my prospect. And I have to say, it's a really exciting area to be in at the moment. Thank you very much, Vicky. I'll stop there because otherwise I'll have run over time.
Brilliant. Thanks ever so much, Tim. That was a wonderful introduction to this webinar. And I think really set the scene of rare disease within the Central and South region. And I think, obviously shows the diagnostic odyssey, but also offers, I think, hope for people in the future of hopefully that being shortened and being less distressing as well. Brilliant. Phil, are you okay to introduce our next speaker, and I'll just share the slides again.
Hello, everybody. So I'm really delighted to introduce our next speaker, a passionate advocate for rare diseases and health equity, Dr. Sandra Butterworth, who is the CEO and founder of RareQual, which is Rare Quality of Life, and ADIRA, which is Equality, Diversity and Inclusive Research Association. Dr. Butterworth, are you there? Hello? Have we got? Hello, Dr. Butterworth, Sandra. I did see. Hello, I can see you in the chat.
Hi, Sandra. I've seen that you've just replied.
I wonder if the microphone's not working properly.
Perfect. We can hear you now.
No, I can't hear.
Yeah, Sandra, I can hear you.
Okay, so I don't know if anyone else can, because I can't.
I can't hear her. It's Charlotte. I can't hear her. I wonder if, Sandra, I wonder if you need to switch it off, your mute off and on again. To see if we can hear you. Apologies. Oh, thank you. She said she's going to come off again and join back in.
Okay, great. Thank you. I hope whilst we're waiting for Dr. Butterworth to come back on, it's just to encourage you all for any questions in the chat. But I think we can all agree that that was a fantastic talk by Professor Barrett. So hopefully we'll have Sandra back in a second. And I'm sorry, I'm just checking the joys of technology.
It does seem that Sandra's in the chat, Phil.
Okay.
And microphone's on.
Sandra, are you back?
She's back on, but I don't think we can hear you again.
I wonder if it's possible just to swap our agenda around, if that's possible at the moment. I'm just conscious of time and running over. Good. So if we can swap, Sandra, if you can hear me, if we can put you through for the 12:55 session in the meantime, and just try and sort out the our sound issues, and I can send you another link. Is that okay with you?
Yes. Yeah, Helen volunteered to go first, if that's
Sorry
okay.
about that. Sorry about that, everybody. But I will.
Thank you, Sandra.
Try and contact Sandra.
Thanks, Bill. In that case, I'd like to introduce Dr. Helen Stewart, and she's also accompanied today by Amanda and Richard Cole, and also by Karen Bradley, and they're all parents of children who have been diagnosed with rare diseases in Oxford. So Dr. Helen Stewart is a clinical geneticist, and she works in Oxford, and she also works for Central and South Genomics. And Amanda and Richard Cole and Karen Bradley. I think, Helen, if maybe Karen goes first, is that okay?
That's okay. What I'm hoping is that all three of us will put our videos on so that you can see us all.
Yeah, that's great. Thank you.
While hopefully Karen and Amanda and Richard are able to join in, just sort of by way of introduction, I'm a clinical geneticist, and I've worked over the last few years particularly focusing on rare disease and thinking about training and education, doing lots of work with our colleagues in paediatrics and other specialities to teach them how to do genetics. And Tim and I are of a sort of similar vintage. We've both spent the last 25 years or so in genetics and have seen how things have changed, really from a medical and a lab point of view. But what I'm hoping to do today, because it is Rare Disease Day, is to have a conversation with two families who have got children with rare diseases, so that you can hear from their point of view what their experience is of how they've come to have a diagnosis, and really to focus on what it's meant for them, so that you can understand a little bit about the patient's point of view, because that is really what we're all here for. So I can see, I think maybe that's you, Amanda, with a slide that you've put up. Have you prepared some slides? Would you like to just go through those? Or is that Karen? Is it Karen maybe first?
Yeah, that's right. It's Karen first.
Karen. So what I was thinking that we would do is to hear a little bit from both families about how their diagnoses were made. So we'll have the two families, and then we'll have a conversation with both of them about what it's meant for them, if that's okay. So Karen, I'll be quiet and I'll let you go ahead.
Oh, thanks. Yeah, thanks for inviting me to talk. Yeah, I thought I'd just explain a bit about our family's connection then to the genetics team and our experience with the diagnosis. We're actually quite lucky compared to a lot of families that we've connected with since the diagnosis, as in we've had quite a relatively short diagnostic odyssey of about 3 years. I had a normal sort of pregnancy and birth. Had what I thought was a typical child, but I'd already had a child before that, so kind of was used to, well, thought I knew what to expect. So I had a very, happy baby, but he wasn't putting on weight. So the first sort of step really in all of this was that sort of really didn't add up, because there were no other sort of signs of why this would have been happening. But he was diagnosed with a VSD at 3 months, and he had open heart surgery at 4 months. And so we thought that was sort of the end of hopefully any sort of drama around that, but obviously that was just the beginning. Little did we know at that point. Following the heart surgery, he was diagnosed with hypertonia, and I think probably common to a lot of families, he wasn't meeting the milestones. I think at that point we thought it maybe was connected and he would sort of catch up. It became sort of apparent as we started going through this journey, and he had hypertonia, and then we referred to physio, and I started doing a bit of my own sort of research, that I realized hypertonia was also linked to quite a lot of conditions, but nobody had really talked to us about anything at this point. Then sort of around a year, I think that the global delay sort of was mentioned when we realized that so it was everything then obviously that was delayed. And then the sort of big shock really was he had a febrile seizure at about sort of 12 months, which we weren't too worried about at the time, because a lot of children do have those, and he was ill at the time. But then at 18 months old, he had his first proper seizure, which obviously was very frightening for us as well, not really knowing what was going on. At that point, like I said, our diagnostic odyssey was quite short because we'd gone straight in with the heart problem and we were under a cardiologist. We had automatically then seen a paediatrician who was going to sign us off, but of course these things started sort of cropping up. So once we hit the sort of seizures and they started, that sort of prompted further testing, and that was when we had the epilepsy gene panel done. So the results did take around 18 months. I think Dr. Stewart knows it was during sort of COVID when she finally called with the results, and you know, I can't really explain what that really felt like. It's quite emotional when you've been waiting to find out, not really knowing if all these things are linked or how they could be linked or what to expect next. And yeah, our son was diagnosed with HNRNPU disorder, which is he has a deletion in the HNRNPU gene, which is on chromosome 1. So the HNRNPU has multiple roles in the genetic control of our sort of development and functioning. So most children have developmental delay, moderate to severe learning difficulties or intellectual disability, seizures and epilepsy, language and speech difficulties and hypertonia. And then there are other, some children have other symptoms. So Marcus's heart condition was part of that. So that really kind of thrust us into the world of therapies, hospital visits, lots and lots of different clinicians involved, the world of SEN, then following on the world of EHCPs. And if anybody who's in this world as a family in the SEN world knows, lots and lots and lots of paperwork. So I don't know, Helen, if I put together this slide, I was just going to sort of use this to explain what the diagnosis sort of meant to us, but I don't know if you want to hear from Amanda first about their sort of journey.
That's all right with you. Should we do it that way around? And then I'm so conscious that what you said to me in clinic was just encapsulated everything that I wanted you to try to convey today. So if you would like, if you're happy just to sort of hold that thought, and if I can bring Amanda and Richard in.
Yes.
Amanda and Richard.
Can you hear us?
Yes, we can.
Hello, I'm here too.
Hello, both of you. So thank you very much for your time today and participating. So would you like to tell us about your story of how you came to have the genetic diagnosis?
Yeah, sure. So what is quite remarkable really is hearing the previous speaker talk about her family's journey and just how many similarities there are with ours, which I think also kind of just flags that there are so many commonalities between what are a huge bunch of rare disorders. There are some quite specific overlaps, and therefore I think just the recognition of all of those kind of key red flags needs to be better better recognized kind of in the general practice and in the clinics that we along our 5-year path have been through. So to kind of just talk you through our journey. So Ben was born in March 2019, relatively normal birth, but it became quite apparent quite quickly that there were things that weren't quite right. He had quite low muscle tone. He was born an average size and weight, but he had a lot of trouble feeding. We didn't really know why. At 6 weeks, we had a nearly catastrophic episode where he contracted viral meningitis and bronchiolitis at the same time. He became very poorly very quickly. I was in an A&E waiting room with him, and he stopped breathing and had to run him down the corridor, and they had to resuscitate him. And that was kind of a key big event, really. And after that point, when we continued to see issues in him generally being slow to reach milestones, it was often kind of attributed to that episode. And so for a long time, that was sort of the narrative. That was kind of our explanation of why he was struggling to meet the milestones that you'd expect. But a lot of symptoms that we now know are associated with the diagnosis that we now have. He had macrocephaly. We were measuring his head circumference every week for months and months. They were concerned he had hydrocephalus, so fluid on the brain. He was very susceptible to infections. I think he was hospitalized 12 times before the age of 2. He had a couple of brain MRIs. Some anomalies were found there. But it took 4 years for his neurologist in the end to think of doing a genetic test. So she raised it first, and we were like, yeah, that sounds like a good idea. 9 months later, so December 2023, is when the results came through, and they explained that Ben has a neurodevelopmental genetic condition called Milan syndrome. It's from a tiny spelling mistake, so a de novo mutation, which means it's the first seen in him, so he's not inherited it from us. Small spelling mistake in the NF1X gene, and that's really important for the protein that supports muscle development, brain development, and that really tied all of what seemed like a lot of unlucky stuff together. So Ben, and characteristic of Milan syndrome is intellectual disability. He struggles with communication a lot, fine and gross motor skills, vision problems, among other things. We also know that he's quite likely to develop epilepsy. And yeah, the condition is ultra rare. There are around 350 people diagnosed with the condition worldwide. And yeah, love to talk about the benefits of what the diagnosis have been for us. But Rich, is there anything I've left out of the journey so far?
Not in terms of the detail, but I suppose in terms of the feeling, it was the first few years were feeling lost, don't understand what's going on, kind of moving from one seemingly unconnected issue to the next. But of course, we do now know they were all connected, and so it was 4 years of uncertainty, confusion, and so to actually get to the stage of getting the diagnosis, cleared that, and obviously budgets are limited, etc. But to take 4 years to get to that point where we were advised that a genetic test might be useful, if that could have happened quicker, that would have put us in a much better position during those early years.
Thank you both, well, all three of you. Thank you. So we've heard a little bit about the journey to get to the point of testing. So really, what I'd like to ask you next is, Richard, you've just told us a little bit about how it's felt not having an explanation, and maybe Karen, you can tell us a little bit about that from your point of view. But can you maybe tell us a little bit about the difference that it's made to you to have a diagnosis? So, should we start with you, Karen?
Yeah. Well, I think I just put these up because these are sort of cover really the key things for us that have, it's made a difference for. I think while intrinsically I knew I hadn't done anything wrong, like I hadn't done anything in pregnancy that I shouldn't. You do have this guilt, this worry that you're trying to you're the child's parent and you don't know what is going on. You don't know how you can help them. You don't know what's next. Every time you get to that point, you think, okay, we've dealt with this one thing. I mean, looking back now, and I thought open heart surgery was going to be the biggest thing that I've ever dealt with, and I'll look back at that now and I think if that had been it, that would have been a breeze. I think I mean, Marcus's condition is also de novo as well. So I can't speak from the perspective of somebody who is a carrier, because I think that probably encompasses a whole nother raft of emotions as well. But just having that answer, I think a lot of parents, like I said, in a way, we're quite lucky. I know, some people, it's years on this odyssey, is the credibility of all these concerns that you've got, that, well, what about this? Well, what about that? And it can be very easy to be dismissed. And I'm not really blaming clinicians per se, because I think it's very difficult sometimes to piece all these sort of seemingly random things together. But I think sometimes you can feel almost you're imagining it, or I know, you're not sort of being heard. So I think a diagnosis immediately gives that credibility. I think provision to therapies and services is a massive one, because there are I mean, obviously things like EHCPs and support services are supposed to go on need. You don't have to have a diagnosis for that, but any families living this know that a diagnosis makes things so much easier to access. We had a nearly 3-year-old who couldn't walk and talk, and yet when we applied for an EHCP, which is Educational Healthcare Plan, the local authority, applying to assess for one, the local authority said, "Oh, we can see he's got some additional need, but it can be met with the local offer." Well, I mean, that's just a load of rubbish. And I think what happens is there's a bit of a wait and see approach, which, understandably in some cases is the right approach, because some children just have a delay and they do grow out of that. But when you have that diagnosis, you know that that's not going to be the case. And I think it also helps with coming to terms with that, because there's a grieving process as well that you go through. And I think when you have a diagnosis and you've got a better idea of potentially what you're going to come up against or what the future may be, you can go through that process as well. I mean, it's a constant process, to be fair, but I think it helps with that emotional side. And then I've put, things like access to support for DLA, and also for Ascend School. So our child is now at a special needs school, and he's thriving. It's a brilliant school, but I think sometimes without a diagnosis, it's a sort of, right, let's put them in mainstream with a bit of support and they'll be fine, especially with budgets and resources being so limited. The other thing for us is with our gene, we have a specific community group for that gene. I mean, ours is ultra rare as well. At the time when Marcus was diagnosed, was sort of 80 odd people worldwide known. So, I mean, that's growing all the time, especially now more people are being tested. But that is a really important part of it, actually, not just for families, but for clinicians, because often as the parent, you are the expert, you become the expert in this condition, because there might be a few geneticists who are experts in this condition, but the majority of people you see, paediatricians, GPs, they don't know anything about this rare disease. So you become the person in helping to inform potential treatment pathways. And actually, it's a bit like it's using sort of live that cohort that you're with, that sort of their experiences to help inform what you might do. I think especially in our group, we've got a number of children who have intractable epilepsy, and people are on different combinations of medication. And actually that experience, you can go back to your neurologist or your paediatrician and talk about, what about this? Somebody's doing this. And I think it really helps with that conversation. And of course, it alleviates isolation, because you do feel isolated. A lot of your friends are not going through this necessarily, or you might be in a group that's just very general, like children with development delay, but actually having your own specific group brings a lot of support. I think, to give an example as well, in terms of proactive treatment, we have a great neurologist at John Radcliffe in Oxford, and actually after Marcus's diagnosis, when we went and saw him, he said, "I've read all of the literature. I know that kidney issues can be one of the issues. Have you ever had a scan?" I said, "No." I mean, we'd had scans for other things, but not that. So immediately that was sorted out. So having that proactive approach, I think, is really important. And for families, I think, like Amanda was saying, if seizures are part of that profile, when something happens, you immediately think, right, okay, I think this is what it is. You've almost sort of, you've got that preparedness as well. So yeah, I think obviously family planning for us, that Marcus was our second child, so we weren't really planning anymore. But as it was a de novo mutation, that did bring some comfort, that if we did want to, that it wasn't something that we had to worry about passing on. And I think finally, I just was going to say that in terms of research, I think the benefit there goes both ways. Once we had a diagnosis, I did a bit of research and found the geneticist who specialises in Marcus's condition and linked in with her. I then linked her to a group in the US that I'd found, because we have an umbrella group for our gene, the HNRNPs. It's a sort of group, and there's lots of genes underneath that. And then we now have a conference every year, which includes clinicians and patients. For our specific gene, we've set up a non-profit, and we've been fundraising, and we're moving into that sort of clinical trial space. So that benefits not just families, it benefits clinicians, the NHS, who obviously we're very aware of limited resources and budgets. So I think family and patient advocacy in this area is really important, and we've participated in a number of studies. So we're doing a UCL study on neurodiverse children who have difficulty developing language. We're now just part of the Genomics study from Bristol, improving the treatment of people with rare genetic diseases. We've been part of a natural history study for our gene, and giving samples has also led to an epigenetic signature as well for our gene, which obviously helps clinicians. So that probably sort of covers it, but yeah, I don't think people always realise the far-reaching implications of having a diagnosis, and yeah, there's so many elements to it, and hopefully I've just given a bit of an overview of those.
Fantastic, Karen. It's a really moving and eloquent description. And the words I've written down as I've been making notes, I wrote down far-reaching. And I think that that's a really important point to get over to people, of how widespread the ramifications are of having a diagnosis. I think sometimes people think, oh, well, there's no particular medication that's going to treat a particular condition, so what's the point in making a diagnosis? But I hope people have heard loudly and clearly that there are very, very important implications for making diagnoses for people. And I think it's our job as clinicians to help facilitate those things. So moving on from that, back to Amanda and Richard, if you could tell us a little bit about what the diagnosis has meant for you as a family.
Yeah, thank you. And thanks, Karen, really, really articulate. And you basically took all my talking points as well, which are very, very similar. But yeah, just echo everything that Karen just said, really. And it makes thinking about all the newborn screening activity and the generation study is an absolutely fantastic step forward. Obviously, it's a subset of rare diseases, and they're the ones that have treatment options. But as you've just heard from Karen, I think just understanding the benefits of a diagnosis, even when there aren't any treatments yet, is really important, and important to have kind of a general awareness of across the health service, and to make kind of clinicians think quicker about the potential for a genetic diagnosis to provide some answers where there are unanswered questions. So yeah, as I say, for us, very similar list of benefits. We now have access to the right medical teams and tests to keep an eye on symptoms we know are associated with Milan syndrome, and that could get worse over time. And that's kind of unlocked a lot of new specialists for us. So we now are seen by a cardiologist to look for known potential complications. Ben has just received a certificate of vision impairment after having seen some new eye specialists. And that means that we can access various kind of local supports around his vision. He's had a specialist teacher for the visually impaired visit him at school, and they've been able to provide advice to his teachers and his the people he works with there. So yeah, unlocking the disciplines and the advice that are going to help us and help Ben kind of reach his potential. It helps us better plan for the future. We have more information on what support he's likely to need in the future. It gives us kind of the language to talk about and explain Ben. It was really hard before we got the diagnosis, when you're talking to friends or family, and especially when you meet new people and you're trying to give them some context about your son without having the words to be able to describe them. It was quite difficult. Having now kind of the language really, really helps in that regard. yeah, the evidence and the specialists helping him now in the education setting have been really, really important, and feeding into his education, health and care plan as well. And like Karen said, kind of tapping into, but also contributing to the active research to advance our understanding of the disease, of the science. We're really lucky to have as well a foundation, a Milan Syndrome Foundation, which kind of connects families, fundraisers, brings together medical experts. They have a biannual conference, which I attended in the US last summer, which was, yeah, just amazing, and all run by families. And we're very, very early on in the research space, but to be involved and to be able to contribute to that is really important. So I'm a health economist by training. So I'm a researcher, and I work a lot with pharmaceutical companies, and I understand, you know, incentives for innovation, and the power of data. And now we have a diagnosis, we're able to kind of contribute our data, Ben's data, to you know, advance the research to understand natural history, which might kind of one day become what unlocks or enables the research that may one day lead to treatments. Rich, do you have anything to add?
Yeah, once again, I mean, yeah, everything Karen said, we could have said as well. It's a very, very similar story, seemingly, which is surprising given that they're obviously very different conditions, but the same experience. And then Amanda took through very practical benefits of getting the diagnosis and how it has helped us in material ways, and helped on the medical side and with the educational side. But I suppose I would just add to that is on our emotional side as well, our ability to deal with this as parents, having gone through a very uncertain time, to get a diagnosis, to understand what's going on, to know that it's a genetic condition, that realistically this isn't going to change, and so this is lifelong. Whereas before it was uncertain. Before, maybe we had a bit of false hope. Maybe he was just catching up to that early episode he had at 6 weeks. Maybe it was all to blame with that, and eventually he'll get there. Whereas actually this gave us the information that no, black and white, this is just who he is. We've got to accept that. Now, that's quite brutal, very hard to go through that process, but we have been through that process now and come to a level that we are more accepting of it. We can get on with our day-to-day lives very happily now. We just know this is what Ben is. And so that, for me, has been very powerful as a way to allow me to actually get back to focusing on life and getting on, and not just this becoming all consuming. Amanda also mentioned about the having the language to speak to others, to speak to people you meet, to people in education, healthcare professionals, but also I'd add how to explain Ben's condition to his sister, our daughter, who our little girl, who's 2 years older than Ben. We could never really explain why Ben wasn't speaking. Her friends would be asking him, so why doesn't your brother speak? And she would go, don't know really. Whereas now we've been able to explain that to her. We're quite open about it, and she's accepting of it. And so for her as well, she's hopefully going to grow up just accepting this as a normal thing, rather than this big question mark or something that's always dark and mysterious and a horrible input on our lives. Actually, we can just accept it and have a happy life because of that as a family.
Thank you all three of you, that's fantastic. I think one of the things I've observed with both of your families is the learning curve that we've sort of been through together, that there's been sort of working together, I think, to get to the point of making a diagnosis. And again, I think that's a really important message for doctors who are working with families with children with rare diseases. So I'm just going to ask you very quickly that in terms of what messages would you like to convey to clinicians or the laboratories, other families, and maybe policymakers? What would you like other people to understand? I know I've sprung that on you. Sorry.
Greater access to testing, earlier access to testing. If there are to maybe the paediatricians who are looking at a long list of seemingly unexplained occurrences, something in there, just question that earlier. Well, is there something linking this? Should genetic testing come into this process earlier to have a look? Costs and times are coming down all the time. We understand, and so hopefully in the future this can become a more routine thing. That if a child, someone is experiencing a lot of difficulties, is there an underlying cause for it? Rather than, as I think Karen said, you almost get brushed off that, oh, that's just this, they'll get over it. That's just this, they'll get over it. Figuring out if there is a link, because yeah, as we've all said today, getting the diagnosis is such a big well, it's a game changer for us.
Thank you.
Yeah, and I would add to that. I know the Department of Health has published the next rare disease action plan for England, and I think a key part of that, and what I would advocate is hugely important, is better care coordination. So Ben's care is across 10 or 11 different medical specialties. We have appointments in 4 different counties. The data collection systems don't really talk to each other, so we're often emailing our paediatric consultant with reports from this, that, or the other. And that puts a lot of onus on parents and carers. And it takes a lot of time and energy, and as well, I think it's not particularly equitable to put that much, to rely that much on kind of the effort of families. And I think the NHS is in a great position to be, to be better at coordinating all of this. So yeah, that would be my plea as well, better care coordination.
Thank you. And then I'm conscious we're coming to the end of our time. I can see that all 3 of the women here have all got stripy outfits on. And I just want to flag up to be, oh, and Richard, you have to, as to why. And I think there's this sort of notion within medicine that common things are common, and that you should think of the common things first. But within the rare disease world, there's the notion that if you see something with 4 legs that looks like a horse, it may well be a horse, but that every now and then you have to think, is this a zebra? And so stripes have become a bit of a sort of meaningful, what's the word? They're sort of significance really, to sort of flag up to people that every now and then, and I don't mean to refer to little children as zebras, but the concept that sometimes you need to think outside of the box and think, is there something rare going on? So that's why we're all wearing stripes. I'd like just to thank all 3 of you so very, very much for sharing your stories, and I hope that people have really heard what you've said today, because I hear stories like yours a lot of the time, but for those people who don't hear it directly, I hope it's been really, really impactful. So thank you very much. There's lots of clapping going on. Has anybody got any questions? I can see Charlotte Hitchcock, you had a question. Is there anything that would help you when you end up being the educators for other people, educators of medical professions, etc., about your child, and the sort of concept of you becoming the expert? Charlotte, do you want, I probably didn't ask your question very well.
Yeah, it was just sort of thinking about the fact that a lot of the time parents I have heard from other parents that we have within our PPI group, that they sometimes feel they spend a lot of time educating about their child's condition and having to explain it to medical professionals, to schools, to other people. And so they stop being so much the parent, but start to become more of someone who is there to educate. And I wondered if there's anything that we can do within our education of healthcare practitioners, or designing of anything which might help when you are actually going out to schools. I know Karen said it's really helpful with Unique, that they are very supportive, but I just wondered if there's anything at all that you feel could be done by people such as us when we're educating and looking at how to interact with all communities.
It's a tricky one because I hadn't sort of necessarily thought about it, but I will say I think there's not a standard as well, sort of between clinicians, that I suppose it would be great if there was a bit more sort of equity in terms of how in terms of their approach. So what I'm saying is it can really vary person to person with you know the different sort of people that you meet and the different clinicians. So for example, I was talking about our neurologist who was very proactive. Before the appointment, he'd already read up about the diagnosis and so was prepared and had that proactive approach. I can go and see someone else. They haven't read the notes. I'm actually telling them what happened. It's in fact exactly what Amanda was saying in terms of people being joined up. A lot of effort goes from parents in us doing the joining up for the clinicians, because either the systems aren't there or they haven't got the report, they don't know what so-and-so said. And so even in the approach, I think if there could be more consistency, so if you're, and I know it's really difficult because everyone's so time poor, but just before you have if you're having an appointment, and whether this is at the school or wherever you are, if people could take 5 or 10 minutes just to read the background before we come in, it would be, really appreciated, because you do feel sometimes you want to sort of make a little recording and then just play it because you say the same things, all the time. So I think that sort of consistency and yeah, I don't know from what else Amanda and Richard would say. But
I would absolutely agree with that. And I think the point of giving someone a diagnosis as well is hugely important and impactful. And how that's delivered can make a huge difference as well. When we first got Ben's diagnosis, it wasn't with Dr. Stewart, it was with our neurologist. And they were kind of reading it from the screen as we were sitting there. Knew nothing about the condition or the implications at all. They even said, "Oh, I think it's probably somewhere between rare and common." Like they didn't know anything about it, and that left a lot of uncertainty with us. And there was some technical uncertainty with Ben's diagnosis initially, anyway. But I think, just better care, particularly on those big moments as well, to do background research and is important.
Yeah, I think all care in terms of that. I remember when we had the diagnosis of epilepsy, and it was just, right, it's had a seizure, we've got epilepsy, here's a leaflet, off you go. And I thought, well, what about, is he having a seizure at night? What if I don't know? What am I going to do? Nobody, there's nobody there to sort of talk through. So I think thinking about the impact of when you deliver that information is spot on.
Thank you, everybody. I'm just conscious of time. Vicky, I think you wanted me to say a little bit about the generation study. Do you still want me to do that? I've got, I think, one slide. Or have we used up our time?
Yeah, I'm sure one slide will be great. Thank you very much.
Have you got it, or do you want me to put it up?
If you can put it up, please.
Okay. So just to conclude, so thank you very much, Amanda, Richard and Karen. I think that's been really, really helpful. So thank you so much. So I'm just going to... Can you see that? Can you see that slide?
Yeah, we can see it. If you just want to, yeah, that's it. Perfect.
Okay. So just to say a little bit about the generation study. So Tim's already talked about it quite a lot. There's quite a lot of writing on here, sorry, but just to say a little bit more about it. So the generation study is a research study, and it's providing genomic newborn screening. So aiming to do whole genome sequencing for newborn babies. So the process that we're following is to consent the mum during pregnancy, having talked to her about it and explained the process. When the baby's born, the aim is to take cord blood, and if that fails, to take a heel prick, with that very, very much not disrupting the standard of care newborn screening, so the standard heel prick that babies are all offered. The aim is to look for 500 genes that can cause about 200 conditions, all of which are serious and treatable, i.e. there is some intervention, but really emphasising that there's not a promise of a cure of those conditions, but maybe altering the course of that condition. Babies are being tested on their own, so as a singleton. The aim is that the turnaround time will be a couple of weeks, and we're expecting that about 1% of babies will have a diagnosis. So if nothing is found, or if in the small proportion of cases, if the sample
Have we
found...
lost Helen?
Oh, can you not hear me?
Yeah, you're back, Helen.
Oh, sorry, don't know what happened. Where did you get to? Did my Wi-Fi drop out or something?
Yeah, I think so.
It's fine, Helen. I can see where you were.
You can see, okay. So if nothing's found, or in a small proportion of people, if the sample fails, people will just get a letter. But if we suspect a diagnosis, then people will go into a paediatric pathway. And hopefully, in contrast to what Karen and Amanda and Richard have said, the person that will be giving the information will know about the condition and will be an expert in that condition. So they'll say, we suspect that your baby's got such and such a condition, and this is what we're going to do about it. So babies then will have confirmatory testing. So if, for example, we think they've got a biochemical problem, they'll have biochemical testing to establish whether there is a definite diagnosis or not, and then they will start on a treatment pathway. So the aim is to test 100,000 babies. We're aiming to keep going with this until March 2027. It shouldn't disrupt the standard of care testing, so if a baby's ill, they would just get the kind of testing that they would get anyway in that pathway. And while we've been setting this up, we've been establishing processes. Oh, what have I done? What have I done? Sorting out the funding and making sure that we've got all the staff in place ready to do this work. So that all sounds very simple and all very positive, but it's a complicated process. We're working very much with our colleagues in the ethnicity, diversity and inclusion world to make sure that we can provide this as a thing that's provided equitably for people, no matter what their position in life. Thinking about informed consent, if you're testing people for 200 conditions, it's very difficult for people to know exactly what it is that they're letting themselves in for. People will be consenting for their data to be stored and to get a result. So sometimes with research, your information goes off into a black hole and you don't get anything fed back to you. But in this situation, people will be getting a clinical test as part of this research. One of the things that's unusual about this testing is that at the point of the blood sample being taken, so as soon as the baby's been born, that baby doesn't have a phenotype. They don't have anything to indicate whether they may or may not have a biochemical problem or an immune issue, etc. The laboratories that are returning the results won't be returning variants of uncertain significance. So there'll be a lot of automated interpretation of the data, but before the data is returned, a scientist will look at the data before it's returned. I think one of the things to really sort of spell out that single gene disorders are uncommon, that genetics is extremely complicated, that when people have an alteration in a gene, that's not the whole story, that people have got 3 billion letters of the genetic code that are all interacting together, and we don't necessarily understand what modifies a gene. So we don't understand why sometimes a genetic alteration will cause a problem for a person and sometimes it won't, or sometimes why affected people might have slightly different features or sometimes very different features. So if you're testing a child who hasn't got anything the matter at the point of testing, if you find a genetic alteration, trying to work out is that going to cause a problem for the child isn't necessarily straightforward. In this situation, we're introducing lots of tests in one go. We're testing for rare conditions, so even though we're going to test 100,000 babies, it may be that on the list of conditions that we're looking for, we don't find anybody with those conditions. So building up an evidence base for each of these conditions as to whether this is a useful process is going to be quite difficult, I think, from the data point of view. And we do run the possibility that we may suspect a condition in a child who's well, and that child might need long-term monitoring. So I think the simple version of it, I think, looks all very appealing. I think in reality that this is going to be a very complicated process that we're going to need to think through. As I said, this is a research process, so all of what we're doing is going to be studied to try to work out, is this going to be beneficial and should this be an NHS service that's funded in the long run? That's all I was going to say about the generation study. I will stop there.
Thanks so much, Helen.
Yeah. I'll stop the session, so I'm conscious of time.
Brilliant, thank you. And thanks so much, Amanda and Richard and Karen. I think you truly sort of represent the experts by experience that, like Tim alluded to as well at the beginning, and how important it is that you get that diagnosis for all of the sort of, well, for the very much for the future journey. So I'm now hoping that we can go back to, let me just get to the beginning of the slide.
Sandra, can
Brilliant.
you see me?
We can hear you now.
Oh,
Everyone
wonderful.
can hear you. So
I'm going to come off one of them, and hopefully you still can see and hear me. Otherwise, you might get an echo. Yes? Am I good to go? Oh,
yes,
my
perfect.
word. It looks as though I can control the slides as well. Not sure how that miracle happened.
Yay.
So they move do they move for you?
No.
It should have a red box around it.
Okay, that's fine. All right. Do you want me to start, or would you like to introduce? Yeah.
Oh, shall I reintroduce you? So thank you very much. It's Dr. Sandra Butterworth, who is the CEO and founder of RARE Quality of Life, and Adira Equality, Diversity and Inclusive Research Association. So I'll leave it now for you, Sandra. Vicky, you happy to move the slides?
Yes. Okay, thank you so much for bearing with us with the technical gremlins, but that's life these days, isn't it? My name is Sandra, and I'm delighted to be here. Thank you so much for inviting me. And with this session, there's a lot of people online, which is great, but I wanted to try and make it as interactive as I can, given the restrictions of technology. But if anyone has any questions on anything that I say during the session, if you want to pop it in the chat, and I can sort of, chat almost as we go, if that's okay with the organisers as well. So I'm not controlling the slides and the chat. I hope that works, but we'll see. Okay, so the session I was asked to deliver is Challenges in Research Equity. So I sit here with two hats, really. One, I am from a rare disease family, and hence the name Rare Qual, so Rare Quality of Life. So I'm one of 8, and I've got lots of cousins and everything, so we're a big family. And we have a number of different rare conditions in my family, and things like, as well as the rare conditions complicated by asthma, I've got that today, and diabetes, thalassemia, rare cancer, believe it or not, sarcoidosis, vitiligo, all sorts of things, okay? Even my children were born with extra fingers, as were their cousins and things. So there's a lot going on in our family, but only one of us, only my nephew, has ever been involved in any genomics. We just don't do it. Neither do my cousins or anybody that I know in my community. And that's strange, isn't it? But that's the reality. So it's sort of my quest, really, to sit outside of the patient sort of and caregiver or family setting or Black community setting, and sit in the community psychology setting as a mixed methods researcher. And so the other aspect of this is around a call for inclusivity in methodology. So that's what I'm going to be talking about today. So could you just move the slide on, please? Is it moving? It's not. I'm moving it on. It's moved. Okay, it didn't move for me, but that's great. Okay, so challenges in research equity. So at this point, hopefully you will have had a copy of these slides, so no one needs to ask for it, but you can have a look anyway and just have a think, really. And I wanted to be able to get people to put some of these questions that they may have, pain points if you like, into the chat. So I've broken down the title of the session into 3. So the title is Challenges in Research Equity. So challenges. So I'm going to ask, and if people can put it in the chat, what are your challenges? What are your pain points? So challenges, general definition is barriers or difficulties that hinder progress. Progress in what? In recruitment, in keeping people on clinical trials or whatever. Okay, research, and I'm going to talk a little bit more about research later, but research is a systematic investigation aimed at generating new knowledge and hopefully to improve practice. And then equity. So this is a broad definition, as I said, the principle of fairness in access, opportunities and resources, ensuring that all individuals, regardless of background, can benefit from scientific advancements and healthcare. So my question to you, what are your challenges? Those of you who are online may be involved in recruitment of maybe diverse communities or people who are underserved. I live in a rural community as well. So there may be people that are trying to get people to come in or to stay and remain on clinical trials or whatever. I'm just wondering if there are any. So in the chat, thank you very much. I've got cultural beliefs can be a barrier in taking part in research and genomics research. Yes, I absolutely agree. And I've worked quite a lot with different people from different ethnic backgrounds. And very often it may be that people don't necessarily want to know what the genomics or the genetic testing results will be because it's in God's hands. So they don't want it. Suspicion with communities about research. Yeah, and suspicion, and there's a link with trust in terms of that. So there's a lot of sort of historical issues around research and mistrust. So it could be particularly around not getting consent and undertaking tests, taking blood tests from people and then using that data without people's consent. There is a lot of historical evidence of that. So I've got another one, poor genomics literacy or health literacy. Yes, and I've done some work around that where people, particularly, you know, there's a lot of people in my family, actually, we've all got diabetes, but and in the Black community who know everybody gets diabetes, but we don't really know what it is. And, you know, like my dad, he had diabetes. He still loved his rum and he carried on because he couldn't understand what it was and why he wasn't feeling well. So there's a lot of that goes on. Oh, we're going on and on. Time, stigma from the community. Yes, that could be it as well. Oh, there's a lot going on in here. Wider family implications and literacy levels is low in the UK. That's interesting. Yeah, so there are quite a lot of challenges around having to address some of these issues. Okay, so just going to go on to the next slide. So this slide relates to an event and some work that we've done. So ADIRA, which stands for Equality, Diversity and Inclusive Research Association, is a project that we have been doing for a couple of years. And it's alongside the University of Chester, one of the professors there. And we basically wanted to be able to look at sort of issues around why underserved communities do not get involved in various different things related to genomics testing, research and consultations of any kind. And so with the research we did, we've published some papers as well, and we're writing a book, yay, around this. But one of the things that I've been looking at, and I'm doing more and more research into this, are levels of trust. Excuse me. So I've taken a sort of basic sort of socio-psychological model. Bronfenbrennan, I think is the reference, excuse me, and its ecosystems, but basically looking at 3 levels of trust. So starting with the basic level, if you can look at that, in the middle, the individual. So you can pop in the chat, have a think about the individual. What is it about the individual? Who are we talking about? So from this perspective, it might be that the individual is the researcher. What is it about the researcher that the people who are to be recruited don't trust? Is that the issue? And also, there's something else that I have been looking at, excuse me, is the trust success ratio. So it may be that the more trust there is, the more potential for success. Yeah. And so thinking of the individual, the person, the messenger, the person who is talking about the research or recruiting for the research, very often the messenger is the message. So it's quite a lot of work that we've been doing at RareQual is we're being asked to recruit for different clinical trials or for different activities. And there was one organisation in relation to Parkinson's. They got funding from the Michael J. Fox Foundation, and they approached us and said that they have been trying to recruit people from Afro-Caribbean or Black ethnic backgrounds, if you like, to be able to be involved in testing of trials to do with the Parkinsonian tremors and so on and so forth. And they were unsuccessful in recruiting. And they wanted us to do that. And what was interesting is, for me, I just made a few calls, done. But that, you know, people know me and like to think that they do trust me. But the challenge was I couldn't just say, contact those people there, because they kept coming back to me to find, we don't know these people, where are they from, what are they going to do with our data, are they profiling, who are they going to give our data to? So all the usual questions tended to come to me. So when people ask me, will you just share this with your network, it's not a case of just, because that network, many people from ethnic minority backgrounds and communities have gone offline, gone dark, but I know where they are, because there's so much hatred towards people of ethnic minority online that a lot have come offline, so it's difficult to find people. So that individual level of trust is quite important in this current climate that we're in. And then you've got the MISO, though, so the sort of trust in the system, if you like. So the consent, as I said, the data protection, are you doing a community profile? And also things like the system, where do you go? You can't just assume that everybody's details are in a GP surgery about their rare conditions or whatever. I know I didn't get diagnosed until I was an adult, and there's a lot of my family don't know, you know, had a little diagnosis, never seen a physician since. So it, you know, it's the system of knowing, can the people trust the system that the researchers are working in? And then the next level up of trust is the institution. So different institutions may or may not have more trustworthy histories, let's say, and so people may not trust that. And I think many people who go through sort of recruitment and everything don't necessarily understand or recognize that the oral history that is passed down generationally is alive and well. So it may not be written in or taught in the medical books, but it's there. It's very, very active, passed down from, you know, my great-grandfather, my grandfather. We know stories that they have told us because they're just all passed down, a bit like roots, to be honest. So that's an important thing to consider. Let's see. So there's some comments in here. If you're surrounded by people who don't have a shared background or culture, you might feel like you don't belong. Well, that's interesting. A lot of studies don't have funding for interpreters, yes, and or for PIS. I don't know what PIS is in language other than English. Yes, I agree with that. Okay, can we have the next slide, please? Okay, so barriers to participation. Another piece of research that I did, where we that was in 2023. What we wanted to do with ADIRA was consider barriers to participation or barriers to inclusion. And what we did was we did a small survey, which we sent out to different people. It was mainly for rare conditions. And we sent the survey out asking people what are the barriers to inclusion, and we sent it out to the rare condition people living or family members with rare conditions. And then myself and the professor from the University of Chester, we felt, oh, this is enough for a conference. So we had a conference, and at the conference we decided to take ethical approval for the conference, because this is an approach that I feel is perhaps lacking in terms of having the robustness to patients' lived experience, to analysing patients' lived experience, to have two types of data sets sitting alongside each other. And so here I'm asking the question, which would be great if you could put it in the chat, it'd be wonderful if you've got any suggestions. How do we address barriers to participation? So on the right hand side, in the centre, the one thing that was highlighted through all of the barriers was trust. It kept coming up time and time again. But there were things like access. Look at the top above that. Sorry, start with number one. I'm sorry. Psychosocial. So things like fear and anxiety, depression, all those factors can affect people and their desire to be involved in various different studies or whatever. Access to different communities. Language. So that's just not just about difference in language, but it could be the complicated language that researchers often use. So lay summaries are wonderful as an example. So we've got from Tony. Hello, Tony. Need to reach the trusted leaders in communities. Absolutely. That's me. And then cognitive ability is another one, number 4 on there. So there are people who may not be able to give consent. You have to get assent through their caregivers, if you like. And then economic status. If a study is in the other side of the country and you say, we'll reimburse you, people have to have money in the first place to afford the £125 train fare and hotel accommodation. So reimbursing after is not always going to work. Then funding has already been in the chat around funding, that when doing ethical approval or applying for ethics or funding for courses, funding for research, I should say, the cost of inclusion, cost of participation of PPIE is not always included in there, and it needs to be. Disability. So if someone has mobility issues and you're asking them to go back and forth, just pop down to London to be involved in this on the tube in a wheelchair. No. Okay. Cultural competence, which came up as well, having an understanding of so I work with people from Muslim communities, for example, and they want assurances that you haven't used any sort of pork in the development of anything that's going to be used in the trial. Can you prove to them that you haven't done that? No, you can't, and they're not doing it. Yeah. And then intersectionality, which I'm going to talk about in a second. Okay, so just going to some of the chats. Oh, there's a lot on here, which is good. Let's see. Be transparent to communities. Do you know how important that one is? It absolutely is. And I don't know with how many of you or who of you are involved in research, but very often you get situations where one set of researchers will come, we would just want to find out what this is. But another set of researchers came to the same community and asked the same thing 6 months ago, and 6 months before that, and 6 months before that. And the same types of researchers are coming and asking the same communities the same issues. So it's being aware that, you've got to be transparent about the purpose. Another thing I should say that is language, which frustrates me a little bit because I get asked a lot. This is how you can help us, Sandra. This is how you can help us to get more money for our studies. This is how you can help us to keep our jobs, to do more research. This is how you can help us. But then when I flip it and say, well, are you able to help us? Oh, no, we've no funds to help you, but this is how you can help us. So the way that people use the language to go out and recruit communities, it is important because people won't necessarily respond to requests how we, the poor, can help you, the rich. It won't necessarily go down that well. But I'll move on to the next slide because I know I've got a lot to say. Okay, so just understanding of intersectionality. I'm not sure how many of you have an understanding of this, but it was in one of the last slides in relation to one of the barriers. Okay, but it comes up quite a lot. So if you can think of intersecting factors that reduce the equity. It's about round health equity and it's socioeconomic status and so on. So if you have, for example, someone who has a disability, so it's their mobility, so that's a barrier to start with. And then they may have a different ethnicity. So you're opening clinics when it's their public holiday for their ethnicity. And then on top of that, they may be a particular religion, and then you're not having an understanding of cultural competence. So all those overlapping factors can reduce the opportunities for that particular person. I hope that makes sense. Okay. And so what I'm trying to do is thinking about the rare diseases framework. I know it's already been mentioned, but one of the themes that I sort of like to pull out to try to enhance is the theme of the patient's voice. So thinking of the patient voice, I just want to be able to think, could we enhance the patient voice and improve the equitable access by having different approaches? But in order to do that, we've got to be aware of the intersecting factors that can quieten that patient voice. I hope that makes sense to people. Okay, just move on to the next slide. Okay. So when I was doing quite a lot of research around quality of life and biopsychosocial approaches, I wanted to be able to bring this in as a shout out, really, and a call for the inclusivity in different methodologies. So within the world of rare diseases and genomics, absolutely the focus is on getting cures and treatments. That's absolutely the right thing. Okay, so that's not, I'm not saying don't do that, but what I am saying is that we as human beings are not just physical beings. We are psychological and social beings as well, and the other factors can affect can come into play. So we've got all the biological factors that everybody knows. Don't need to go into the medical model that is dominant within this space. Okay. However, there are psychological factors such as people's mental health, coping, stress, depression. There's a lot of psychological factors that underpin people's conditions, and there are a lot of conditions like rare skin conditions that are affected by stress. Yeah, and there are other conditions the same. So it's bringing into play the other factors that are very important, and they're all part of the sort of EDI health equity space. The other factors like the social determinants of health, people's homes, living in damp and poor conditions, cultural competence, which I have already mentioned. So I'm basically saying that the approach, when we're putting together, if you're thinking, I need to approach this particular group of people who are underserved or that are not necessarily represented in this space, you need to be thinking more than just the physiological, I need to get the numbers up. There may be other factors, other reasons why the numbers are not there. And those reasons may be social factors. They may be the psychological factors. People are absolutely frightened to death of going into hospitals. So talk to them when they get there. Get some volunteers to greet them. Things that are not necessarily costing hundreds and thousands of your budget. Do you know what I mean? But it's knowing what those other factors are. Ask the question. It's knowing what those other factors are in order to address them. I hope that's making sense. Okay, so can we go on to the next slide? So leading on from that, in terms of the methodologies, so I'm a mixed methods researcher. So when I was doing my PhD many moons ago, whatever, they said, don't do mixed methods because it's hard. Yes, it is, but it does allow you to see both sides, both worlds. So yes, I understand the qualitative analysis that you can do. You can do linear regression. You can get all the statistics and you can identify and look at the significant differences between this data set and that data set. Fabulous. Fabulous. This is how we can do all these predictions for genomics and trends and so on. We live in that space, so we get that. Okay. So what if then, running alongside that, you've got the patient's stories. You had some really passionate and really wonderful stories about the patients earlier today. And I did some research with Medics for Rare Diseases. They asked me, because I understand how to do quite in-depth qualitative analysis to interpret words. So I treat words like cells in a test tube is the best way to describe it. So if you imagine you have someone on a trial, and they have all the biological testing, so you know what the blood results are, blood pressure, whatever the tests, blood samples and things that you take, so you know that they're taking a drug which is going to change something. So if the patient then is keeping some form of diary, oral diary, picture diary, written diary, whatever, and they're identifying, say you had 75% of the patients are saying, I'm waking up in the middle of the night with heartburn. It's not necessarily on there, or I'm having restless nights or whatever. It may be something that you could cross-reference with the drug trial that people are on. And it might indicate something else, like a red flag. So when I was doing the research with Medics for Rare Diseases, shout out to Lucy, I don't think she's on this today, but we identified the red flags of rare diseases to be able to look at not just the physiological symptoms in the diagnostic odyssey, but psychosocial. So there were things that wouldn't necessarily be picked up, like in childhood. So if children are having developmental problems, it's not necessarily picked up in the biological testing. It's just that someone is having problems at school, that type of thing. Yes. Or people are being diagnosed with mental health problems when it's not mental health. It's something to do with something that's undiagnosed, like Ehlers-Danlos syndrome or whatever, because it's associated with things to do with movement and bone and pain. So basically, all I'm saying is, don't just consider the numbers. Think of the words as well and bring the two together to be able to identify something that might be a red flag. So that's me. That's my little shout out for that. Next slide. Oops. Sorry, my slides there. Oh, was that the last one? No, just bear with me. Mine's flicking. Got it. Okay. Mixed methods research. So this is a little bit of an information slide for you, and I'm gonna finish now. We're nearly on the last slide, and it just gives you some of the context which you've already got with some of the references about that. And then my final slide is a shout out to something that we've been doing. We have had support from a wonderful organisation called Costello Medical. We have an online e-learning platform where this stuff is on a free, guys, until the end of March, a free course on our platform that is going to be a platform for networking and support around EDI, inclusive research, health equity for rare diseases. And if you look at the QR code, it takes you straight to the free course. So, and I hope that people will enjoy that course as well. So thank you so much. I think I've probably run out of time.
Thank you so much, Sandra. Really engaging presentation, and the work you're doing is just so incredibly important. I think we've all gained great insights from this, and I certainly look forward to applying some of these ideas and these thoughts that you've put in my mind to apply them to my work. So thank you very much. I think in terms of time, we're probably going to go on, but I see, is it Vicky? Have you got your hand up?
No, sorry, please ignore.
Oh, right. Okay. Yeah. Yes, I can see that hand. Yeah. Okay.
Thank you very much.
You're welcome. You're welcome. Okay. Thanks a lot, everyone.
Right. Sarah, are you still with us?
I am. Can you hear me
Sarah,
all right?
We can. Thank you so much. Good. And apologies for all these gremlins that we're having today. But it's just such a pleasure to introduce you, another inspiring and leading voice in the rare genomic community, Dr. Sarah Wynn, CEO of Unique.
Thank you so much. And that's such a nice introduction, and I feel a bit overwhelmed about being inspiring, but I will do my best. Could I have the next slide, please? And you guys are quite lucky because you might have realized that Unique has just had a rebrand, if you know us already, and so you're one of the first audiences to see our new logo and all of the associated branding. Many of you might know us already, but we are a patient support group supporting both patients and also the professionals who work with our families. And so all of those who are affected by rare chromosome and gene disorders. Could I have the next slide, please? Like many patient organisations, we're tiny in terms of staff numbers, but we are actually quite big in terms of membership numbers. There's just 8 of us. Many of us are working part-time, and we still have Edna Knight, who was our founder back in 1984, as a parent founder, still on our board of trustees, which is really lovely. Could I have the next slide, please? So I've already mentioned that we're quite big in terms of membership. So although we are tiny here in the UK in terms of staff, we have actually we've just tipped over 30,000 members worldwide. So as you can all appreciate, with rare conditions, having those geographical boundaries in place, it seems like a crazy idea, given that if you get a diagnosis with a rare disease, you're really unlikely to find someone on your street or even perhaps your country who has the same rare condition. So we recognised really early on that we really wanted to be able to connect as many families as possible and provide information as far and wide as possible on these rare conditions. So we welcome memberships from families from wherever they are in the world. We've got over 120 different countries represented in our membership. I've just put the top 10 here. So as you can probably imagine, the UK, because we're based in the UK, has the biggest membership, but we've got lots of members from lots of other countries. With the advent of genomic technology and the diagnostic rate of people going through genetic testing going up, our membership is just increasing rapidly. Could I have the next slide, please? And here is just a sort of handy graph that shows you that kind of exponential raise in membership that we have, which has been based on various things, including, having a website and people being able to find you. But those increase in technologies and the launch of the Genomic Medicine Service back in 2018, and as well as in the NHS, genomic medicine services are being rolled out across the world, which again is great news for patients and families in that diagnostic rate increasing. Can I have the next slide? And right from that first start in 1984, our mission has been the same, and that's really about beating the isolation of rare chromosome and gene disorders. Next slide, please. And the next one. So I wanted to just touch on this quickly, and that is that in the UK particularly, and a little bit internationally as well, there's just been an enormous progress in rare diseases, and I think Rare Disease Day is a really good opportunity to recognise that. So while we often talk about that diagnostic odyssey and people taking a long time to get to a diagnosis, I think it's a really great time to sort of have a look back and see how far we've come. And this first one is the DDD project. So many of our families came through the Deciphering Developmental Disorders project, which was a sort of precursor to the 100,000 Genome Project, in offering sequencing as a means to diagnose children who had severe developmental disorders. Could I have I've got those annoying slides where things keep appearing, which I realise when you're not in control of the slides is actually a bit of a pain. So the 100,000 Genome Project, and I thought it's also a moment to mention that this integration of whole genome sequencing and whole exome sequencing into our healthcare systems is also led to this new project run by Genomics England, which is about using genomic sequencing to as a screening test for newborn babies. And this is a pilot which has just started rolling out across the country. Next slide, please. And this sort of translational research projects have also been sort of backed up by many policies, both from the NHS and at the government level, which again is bringing real hope that rare disease families are being placed at the centre of healthcare policies, and bringing those hopes that more and more people will get diagnosed going forwards. And I've just listed, it's not meant to be comprehensive at all, but just as an illustration of some of those services and policies that are here to help rare disease families like those at Unique. Next slide, please. And I've just mentioned that diagnostic odyssey, and I think it's, I use this slide a lot, and the reason I do is because I think it's a really good visual depiction of what many of our families go through. So at Unique, most of our families have children with developmental delays and intellectual disability. Sometimes they're picked up prenatally, sometimes they're picked up in NICU or PICU, but for many of our families, their children start missing those developmental milestones. They might have a number of medical conditions, but nobody puts it all together and gets that diagnosis. Many of our families have gone through years of testing, different kinds of tests and investigations before they get to that final point of getting a final accurate diagnosis. And I think, although this slide's quite headache inducing, it's really challenging for families to be on that pathway, and often be on that pathway for a really long time. Can I have the next slide, please? So I think often we think about a diagnosis being important because it guides management or treatment. Actually, for lots of families, it doesn't have a magic answer, which you get this answer of a rare chromosome or gene disorder, and then suddenly you get access to a treatment or a specific therapy. For lots of our conditions, which are complex and multi-systemic, the way that our families are treated is the same before diagnosis, where all of the individual symptoms and features are treated, as after diagnosis. So often the answer really isn't about treatment and therapies, but nonetheless, diagnosis is incredibly important. I just showed that diagnostic odyssey, which can be really challenging for families. Families are often looking for a reason why their child has got the features or symptoms or isn't developing as they expect. I think, you know, it's human nature to want an answer, and our families are no different. They want to know, why has this happened to their child? Why has this happened to them? We know often families feel a sense of guilt that it's something they did or they didn't do. So getting a diagnosis can bring peace of mind and relieve that self-blame. We really hope that getting a diagnosis will lead to better information and support for these rare conditions. I'm going to talk a bit about the lack of information and how tricky that is for families in a minute. A big part of what we do at Unique is putting families in touch with other families who have a child with a similar condition. So getting a diagnosis enables you to be able to do that. So you can access information, but you can also access other families who've got a child with the same condition, or might be on the similar kind of journey or pathway as you. We'd really like better research and data sharing about these rare conditions so that we can all learn more and move forward. And of course, the other reason a diagnosis can be really important, particularly with genetic conditions, is it can help families make those reproductive decisions, largely about, you know, if they're looking to expand their family, how they might go about doing that. Next slide, please. A couple of years ago on Rare Disease, Genetic Alliance launched this report. They do a report every day, which is released to coincide with Rare Disease Day. And this is one of my favorite reports, actually, because they went out to the patient community and really tried to look at what made a good diagnosis. So the diagnosis, of course, needs to be the right diagnosis and accurate and be given in a timely manner, but there are all sorts of other components which make the diagnosis a good diagnosis, which means that families can absorb it, understand it, and move forward with their lives. I haven't put the link here, but if you Google it, you can download this report from the Genetic Alliance website, and it's a really interesting and helpful one, so I really recommend doing that. Next slide, please. So I've talked about the importance of a diagnosis, and so these quotes on this slide are quotes from those families who've been on that diagnostic odyssey. So actually, although we might think getting a diagnosis might constitute sort of difficult or bad news for families, we actually see that often families are a bit relieved. So I mean, the feelings are much more complex than that, but that sense of relief is often a bit unexpected, but it is because they've been on this diagnostic journey. They know that there's some underlying cause for their child's issues and symptoms, and they really want to know what it is. So finding out that genetic diagnosis, that molecular diagnosis, can really provide a lot of relief from families. But as I mentioned before, that's a bit more complicated than relief. Can I have the next slide, please? And it can have these huge impacts on families. Next slide, please. And the rarity is a sort of added challenge. So again, I mentioned that there's often not a care pathway or treatment available for these sorts of conditions that we support at Unique. And so it often doesn't provide all of the answer that families were looking for. There's often a lot of a lack of prognostic information and the lack of a care pathway to follow. Nobody's ever heard of these conditions, so there's a real struggle for recognition. Families find it challenging to find support, and they often spend a lot of time having to explain their child's condition to other healthcare professionals, the school, the social worker. And so you can imagine that the rarity itself is a challenge in getting one of these diagnoses. Next slide, please. Oh, if you could do the clicks, thank you. And quite often the conditions that we support are don't have a name, like Down syndrome has really good sort of brand recognition. Lots of people know what that is. The conditions that we support tend not to have that name. They often are named after the position on the chromosome or the gene name. And when families get this sort of diagnosis, what they really want to know is, what on earth does this mean for my baby? How do I explain these results to other people? What's the name of it? They often don't have a name at all. And this is one of my favorite ones, XXXY. This is one of the conditions we support at Unique. And the parent said it looks like a bad hand at Scrabble. Next slide, please. And so all of these things together have really big impacts on families. And I think it's not just about understanding the genetic results, but actually caring for a child with a rare disease has huge and sort of wide-ranging impacts on families. We know that often it means that a parent has to give up work or cut back on work for caring responsibilities, and this can have huge financial and psychosocial impacts on families. We know that often mental health of one or both of the parents is affected. We also know that it affects not just the parents, but the siblings and the grandparents and that wider family who are all trying to deal with this new rare diagnosis. And sadly, in the Unique membership, we do know that many families have split up because of the pressure. But what we also know is that if we can all work together, so professionals, patient support groups, and everybody working in partnership, would really like some of these challenges to be mitigated, and they can really be helped with the right support. Next slide, please. I always mention this because I think it's a really good depiction of daily life for many of our families. It's a comedy, quite a black comedy that was shown on BBC a couple of years ago, and it was written by two Unique members, Sean Pye and Sarah Crawford, and it was depicting the life of their child who has a developmental disorder, which again for many years was undiagnosed. And a couple of years ago, they produced a one-off special after there'd been two series, and that was the moment where they got the diagnosis for their daughter, who in the TV show is called Rosie. And I think it's from both the clinical genetic side, but also from the patient and family side, that giving of the diagnosis, I think, is done not only with humour, but actually with accuracy. Although I will say that the clinical geneticist is sitting behind a desk, and the most common response I get from clinical geneticists is, I wouldn't sit behind a desk to give a diagnosis. So, you know, it's not perfect, but I think it highlights that experience of getting a diagnosis, and then how families then go on to sort of understand and move on from that diagnosis in order to be able to support their child to reach their full potential. Last summer, we ran a huge survey with our unique families, and next slide, please. And we invited them all to answer some questions, and some of these questions were on that general experience, that day-to-day life with a child living with a rare condition. And we were also asking some questions about how we at Unique might be able to support them better, what services worked and what might not work at all. And I'm not going to go into lots of detail of this survey. We're going to publish it in full later this year, but I just wanted to share a little snapshot of our results. We had over 2,000 responses from all over the world. We faced interesting listening to Sondra just now talk about inclusivity and diversity, and it's a challenge for us to make sure that we are inclusive and diverse. And we're not, and I need to come and get in touch with you, Sondra, about what we can do about that and do a better job. But one of the issues that we quite often face is that most of our members are mums. So we are very, very much, so the results of this survey are very much sort of the maternal experience of caring for a child with a rare condition. Can I have the next slide, please? But one of the questions we asked families, and we gave them a list of sort of both positive and negative words, and we asked them, from this list of words below, which best describe your experiences of having or caring for someone with a rare chromosomal gene disorder. And we know from our daily working with these families that the fact that 70% of them said it was an anxious experience was not a surprise. But that is worrying. You know, that's not what we really want families to be reporting as being their primary word. 60% said they were frustrated, and often that's about how difficult it is to access the services and support they need. 60% said they were stressed. 51% said they were isolated. And then the fifth most common word that came up was determined. And I think those of us who work in patient support groups know that our families often have to be incredibly determined and tenacious in order to be able to advocate for their child and to get their child the right support, and in school, in healthcare, in social settings. And so that word determined was also not a surprise to us. Could I have the next slide, please? So I just thought I'd give a quick whistle-stop tour of the things that Unique can try to do to help mitigate some of these challenges of living with a rare condition. Next slide, please. Oh, sorry. Only 2 clicks, you're done now. Thank you. So we've really got 2 arms to the support we offer. We are really much a very strong information resource, and some of you, and lots of you I hope, know that we produce information guides to those individual chromosome and gene disorders. They're all on our website. And can be downloaded and shared as widely as possible. As well as the information guides, we have some practical guides. We have lots of regular magazines and e-newsletters that go out to families and also professionals who are signed up to our mailing list. But the other arm of what we do, of course, is support. And that support is provided both internally from Unique staff. We have a email and telephone helpline. We have a private Facebook forum for our members. So it's a safe space just for the membership of Unique, where they can ask questions and share experiences, sometimes just have a rant. Share and celebrate their child if they might have achieved something that is really meaningful for their child, but might not be so much in the general population. We match families together so they can provide peer-to-peer support that way, and we run lots of events for families to come together, both online, so we can look after our sort of worldwide population, but we also do events dotted around the country in the UK, and we have a couple of events this year in Wales and one in Kent. So we sort of move around to make sure we try to give everybody the opportunity to come together and meet others as often as possible. Next slide, please. So I just wanted to mention, these are the information guides I've just mentioned to different genetic conditions, and of course we have the chromosome ones and the single gene ones. Again, because we are so keen that we've got this information, we want it to be accessible to as many people as possible wherever they are in the world. We have lots of them translated into different languages, and you can also download them from our website. That's done on a voluntary basis by clinical geneticists around the world. We have an amazing setup in Russia, where an amazing Russian clinical geneticist has embedded translating a unique guide into the medical training. So we've got over 100 guides and more coming in Russia, and that's done by clinical geneticists not just in Russia but in Ukraine and Belarus and other countries that speak Russian. And so you know, these are just amazing ways in which we can get this information out far and wide so people wherever they are can access it. Could I have the next slide, please? And the guides are used in the obvious way as about providing information when families get a diagnosis, but we also realise that they have a sort of bigger life than just that. We know that families find that they get this diagnosis they've never heard of, and getting a leaflet is a real validation of that condition. Families look in it and they see their child in this leaflet, and that can be an incredibly powerful emotion for families who've been feeling isolated and confused and anxious. So being able to see others, seeing photos of children that might look like their children, and descriptions of families where their children are doing similar things can just be really brilliant at relieving that isolation. We know families use it as a tool to hand to other family members, the school, the social worker. We also know they get used in tribunals and panels where parents are advocating for support for their child. So we know that they get used really far and wide for sort of multiple different reasons. And I think it's a real highlight of how important high quality, well-respected patient information is. Could I have the next slide, please? I think 2 clicks and you'll be there. 1 more click, that's it. I also briefly mentioned in a previous slide that we have practical guides as well. So although the information on the particular genetic condition is really important when people get that diagnosis, at that moment of diagnosis, and looking forward to what to expect going forwards, actually we know that the day-to-day concerns of our families are often very similar, even though they might have different genetic conditions, and things like sleep and behavior that challenges and communication, and these sorts of day-to-day experiences crop up for many of our families. So we've got lots of sort of much more practical information guides which can signpost to lots of different organizations that can provide really good support for those. Could I have the next slide, please? We are keen to present our information in as many different ways so that it's accessible for different people who might want to access information in a different way. And so we've got a couple of really fantastically cute animations about the techniques and array CGH and DNA sequencing, which is how people get their diagnoses. You know, these sorts of quite complicated technologies have been described in these animations where a cell goes shopping for all the things it needs to be able to do its job properly. And these have been really popular with patients and families. We've also recently developed a series of guides aimed at children so a My Chromosome Story, and this slide here is a new project that we are running, which is producing easy read guides. So these are those really guides that are set out for people with intellectual disabilities, which is a really large part of our unique membership, and we have a number of guides in this easy read format, and hope to have many more coming. And so again, those are all available on our website if you want to have a look at those. Could I have the next slide, please? And the next one. And right at the start, I said that we are supporting all those affected by rare chromosome and gene disorders. So that means people, patients and their families, but we also want to input and support with and work together with healthcare professionals and policymakers. So we take part in lots of national and international research projects at all stages. So from application stage to just sharing research projects with our families. We are involved by in advocating on behalf of our patients and making sure that lived experience is taken into account in as many sort of places as possible. And so me and other members of the Unique staff sit on lots of boards and take as many opportunities as we can to present to policymakers, clinicians and scientists, really anyone who will have us and will listen. And we work really closely with other organisations, both to do campaigning with organisations like Genetic Alliance, who I mentioned earlier, but we also work really closely with the condition-specific organisations. So we're sort of an umbrella group with over 1,000 genetic conditions under our umbrella. And generally, we will signpost, if there is a specific organisation, we'll signpost families there and they can join us and also go there. But when we're producing those information guides I mentioned before, ideally we work with that organisation as well. So we've just published a guide to a condition, and that was done in collaboration with the condition specific support group, and we use information from their members as well as our members, as well as the published medical literature, but then we co-brand, and so then the information guide can sit on our website and their website. So we're really keen to be as inclusive as possible and work with everyone so that that information is the sort of the gold standard comprehensive information on that condition and can be found in all the places where you might go looking for information and support on that condition. Can I have the next slide, please? And I mentioned the events we have that go around the country, and these are some pictures just from some of our events. You can see one is in Exeter, and so we invite all of the families that live in that area, but we also invite the local genetics service, so the clinical geneticists, the genetic counsellors and the lab staff, and so families and the professionals can kind of interact, and they're a really fantastic event for everybody. I sometimes think that the lab staff are the people that enjoy it the most, because often they're working really hard to deliver these diagnoses to families, but actually they never really get to meet these families. So it's a really, really fantastic opportunity for everybody to actually meet in person. Next slide, please. This is actually my penultimate slide, and I just wanted to share some photos and really thank the families that are really the heart of Unique, what Unique's all about. And there's some photos here of some of our families who share their experiences and their stories and their lives with us, and we're really privileged to be in that situation. And I think the next slide is my final slide. And I just want to thank you again for giving me the opportunity to come and talk to you today on this very special Rare Disease Day. And I'm happy to take questions if there's time, but otherwise you're very welcome to email me. And, we're always here to answer any questions or provide any help that might be useful. So thank you very much.
Thank you so much, Sarah. That was just such important information to get out there. And I think I've spoken to you before, and I use Unique when I'm trying to support nurses in understanding. And the first time I became aware of Unique was during the DDD study. And actually, going forward a few years, it's quite incredible to see people still, not everybody is aware of Unique and how and the important resource you are. But yes. Thank you so much.
Thank you.
There's just two comments here in the chat. I think the one is from Amanda Cole. That's the unique guide you're referring to is the one you published last year.
Hi, Amanda. Yes, exactly that one. But we do this this system of working with other organizations is one that we do very often, and we really want to continue to do it because it makes real sense for us to work together and not duplicate effort, basically.
Yeah. And then from Charlotte then as well is that working with the GMSA, I think there's lots we can do together. But those are the only things that we have there in the chat. And thank you so much. And I'm so sorry for keeping you past your time that you're available.
Don't worry. Don't worry.
Really
I know how
valuable
it is.
information.
Take care. Thank you. Thank you. Thank you.
Lovely. Thank you. So we've got our final speaker, and again, apologies, Antonio, but I am really pleased to introduce you, Mr. Antonio Ochoa Ferraro, who is our consultant pharmacist for inherited metabolic disorders at UHB. Are you on the line?
Yeah.
Thank you.
Are you able to listen? I've tried to take control of the presentation because it's probably easy for me. I don't know if that's let me see.
Let's see if that
is
works.
It moving?
Yes, it is.
Okay, yeah. So,
fantastic.
Brilliant. Okay, so well, first of all, thank you very much for inviting me today. And I might be start with disclaimers because I'm going to be talking about some treatments, and because I've worked with pharmaceutical companies, it's worth to declare it from the start. But any payments I will normally donate in full to the UHB charity. So I'm going to talk about rare disease and the diagnostic odyssey, but from the point of view of how I explain it to undergrads and other professionals. And then I'll talk a little bit about the generation study, but I'm planning on giving it a twist to explain where it comes from and the link with phenylketonuria, which is one of the conditions we look after. And then I'll give you some examples of how we're using this newer and exciting world of pharmacogenomics in the world of rare disorders and rare disease. So first of all, when part of my role, I have to do teaching to undergraduates in pharmacy and trainee pharmacists and other healthcare professionals, which up until now, they haven't been explained what rare disorder is and what the diagnostic odyssey is. So I think I'm trying to educate my colleagues, not to be experts in the 7,000 conditions, because that's not possible, but actually to let them know what a rare disorder is. And one of the challenges is most people come from an education where they have been taught that rare diseases are rare and common conditions are common. So obviously a single rare disease tends to be ignored. And there are some challenges also because there is not really a unified definition in what a rare disorder is. The Americans use less than 200,000 people affected, whereas we go for prevalence 1 in 2,000. When you work out the numbers of the population, it's actually pretty much the same, 1 in 2,000, 1 in 2,500. So it's not very different, but we're using different terminology. So it's important to make people aware. It's really something that's not very common individually in the population, that there are more than 7,000 non-rare diseases and more that we need to find out and give them a proper name. 80% of those will have a genetic cause, as we've been talking today. And when we look at the general population in the UK, we're looking at 3.5 million people, or 1 in 17, as it was highlighted in the UK strategy for rare disease. So what people are not usually aware is that just because you have a rare disorder, that doesn't mean that you're going to look different. Most patients actually don't look any different. I'm not sure if many of you would be aware of those celebrities that have a rare disorder. Selena Gomez, George Clooney, Miles Davis, he had sickle cell. Gaten Matarazzo, I think he's more known these days, especially in the younger generation with the Netflix series. But just having a rare disorder, that doesn't stop people achieving high success, winning athletic gold medals. But what I try to explain to anyone who listens is that most of the time, patients with rare disorders don't necessarily know they have a rare disorder. And it is a big challenge for them to actually find out what's wrong. So you can go online and you will probably find lots of different examples of people that you've heard of but have a rare disorder. So obviously we've talked about the number of misdiagnoses, the number of specialists they have to see, and the number of years they have to wait on average to get that diagnosis. And that illuminating reality, that average of 4 years, it's also because of the newborn screening that brings down the diagnosis down to 5 days or 10 days. So moving on to the newborn screening, it is all due to phenylketonuria, which is a rare inherited metabolic disorder. It's a deficiency in the phenylalanine hydroxylase. That's the enzyme that converts phenylalanine into tyrosine. And the patients with that metabolic disorder accumulate phenylalanine in their body, and that, if it is untreated, causes irreversible intellectual damage down to psychiatric symptoms. These patients, when they were not treated, will end up in care, most of them unable to speak depending on the severity, completely dependent, and not being able to have a normal life. If it is treated, that's a different story. And there is a treatment, and it's all thanks to this little girl that was born not far from where I work here in Birmingham, in Queen Elizabeth Hospital. So she was born in a maternity hospital in Lodgewood Avenue that then later transformed into a women's hospital close to Queen Elizabeth. And Sheila was born with PKU, and when she was about 2 years, she was diagnosed by the Children's Hospital. And back in the 50s, the early 50s, that was the end of the story, and there was no treatment. But Sheila had a very stubborn mother that wouldn't accept that that was the case. And she literally harassed the doctors at the Children's Hospital to develop a treatment for her. And they actually developed a treatment. It was an original N of 1, a clinical trial with 1 patient only. There are some nicks and knacks on that, but thanks to Sheila and her mother, all the phenylketonuria patients, all the PKU patients in the world have a treatment nowadays. And unfortunately, she didn't benefit from it. It was, first of all, it was too late for her to benefit from it. And secondly, unfortunately, she was lost to follow up and never actually benefited from the treatment. But because there was a treatment, Robert Guthrie developed the newborn screening test to actually find those patients with phenylketonuria that could be treated early, a few days after being born, and actually prevent them from developing all the neurological damage that will stop them from having a normal life. He actually had a niece that had phenylketonuria and was diagnosed too late for her to benefit from the treatment. And he had a son that was never diagnosed that also suffered from mental retardation. So he was personally invested in getting an early screening program of the ground. But before he started the program, phenylketonuria was thought to be 1 in 50,000. By the time the screening program a few years later was mandated by law in many countries, it was more than 1 in 10,000. So when you start looking at for a condition, you actually find it and it becomes more prevalent. So nowadays in the UK, we're looking at 9 rare disorders. It's going to be 10 with the addition of another one. You can see the different prevalence of them. So the common one that people know is sickle cell, cystic fibrosis, and congenital hypothyroidism. But it was phenylketonuria that started all. And then now it's moving on to the newborn a generation study that is going to be looking for rare conditions and enable to diagnose them sooner. So it's going to be 200 conditions that we've been talking, and one of the key things, and a pharmacist, that's what interests me, they can be treated through the NHS. And some of those conditions we look after here in inherited metabolic disorder is homozygous familial hypercholesterolemia. And this is an image that we like in the world of lipid disorders, explaining that when you reach a certain level of cumulative LDL cholesterol, the one that people call the bad cholesterol, you're likely to have a heart attack. And for patients with homozygous FH, that age is reached at 12.5 years. So it's not 12.5 years old shouldn't have a heart attack, but if you have homozygous FH, it's not uncommon that if they haven't been diagnosed, they will suddenly die because of it. So this generation study will allow us to find those patients sooner, and there will be treatments for them, because we will have, we do have the statins that everybody knows of, and ezetimibe, but there are also some injectables like evolocumab, which is a monoclonal antibody, and evanicumab, that's a new one that just came out. Lomitabide is another oral tablet. They can go through apheresis, which is dialysis to remove the excess cholesterol. That's very demanding for the patient, and they have to go weekly, so in some cases, the more severe ones. If nothing works, they will have to have a liver transplant. But hopefully, if the results of the generation study allow us to diagnose and treat these patients sooner, they won't need to go through the liver transplant. We'll be able to manage and treat them with these treatments that are all available in the NHS. So when it comes to rare disorders, I work in inherited metabolic disorders, so these are conditions where just a single enzyme or protein is defective or absent because of a single gene variation. And it tends to lead to an accumulation of a substrate or the lack of one substrate of a product. And could also lead to the production of a toxic byproduct, trying to eliminate the product that cannot be eliminated through the normal pathway. So either we don't have product D because we don't have that enzyme and we accumulate C, or that goes into something X that can be toxic. So I'm going to give you an example of one of the conditions we look after here, which is Fabry disease, which is alpha-galactosidase A deficiency, and it's one of the lysosomal storage disorder because it's one of the enzymes in the lysosomes. These are the digestive system of every cell, and without that enzyme, you start accumulating that globosyl triacylceramide, and that causes significant problems. Or these are multisystemic conditions because every single cell in the body has these lysosomes, and if they don't have that enzyme, obviously that product begins to accumulate everywhere. So patients with Fabry disease will have problems mainly in their heart, their kidneys, so they're going to have renal failure and require a kidney transplant very early, or they could have cardiomyopathy, hypertrophy. They will have different symptoms, gastrointestinal symptoms, eye changes in the back of their eyes, neurological symptoms, very peculiar skin rash. So, but these are slowly progressive conditions. So this builds up little by little over the years. So it's difficult to diagnose because not all of the patients will present with all of the symptoms, and not at the same time, and it would be difficult for any healthcare professional to have been aware of all of this. So this means these are the patients that don't get diagnosed when they're 10 days old. These normally tend to be diagnosed when they're on their 40s, 50s, and unfortunately, most of the damage has happened already, and the treatments are not going to be as effective. And there are actually 4 different treatments. So for a rare disorder, this is an unusual case where we have 3 enzyme replacement therapies where we can give the enzyme that they cannot produce. We can produce it in a factory, put it in a vial, and administer it to the patient. And there's even an oral treatment, an oral chaperone treatment. And all of these 4 treatments are available in the NHS. Now, I wanted to talk a bit more about the migalastat, which is the oral chaperone treatment that has been available to us since 2017, because this one is recommended for those patients that have what we call an amenable mutation. That's the actual wording in the NICE recommendation. And what it means by an amenable mutation is that some patients with Fabry, the gene variant will not respond to this treatment, but for those who are going to respond, we can prescribe it to. So we have to do some bit of digging, find out what the gene variant is for that patient. And obviously the company is very keen for us to prescribe it. So they make it easy. They allow us to, they have this website where you put the gene variant of a patient, and it will tell you whether that gene variant is amenable or not. And I've gone for that N215S because it's the most common gene variant for Fabry disease in the UK. There's a bit of a founder effect. So we have a lot of 40–50% of our patients will have that gene variant that fortunately is amenable. And what it does, it's kind of counterintuitive. It's an inhibitor of the enzyme. But these patients, what happens is they can't produce some enzyme. So when we measure it in a blood spot, it will have a little bit of activity, but most of the enzyme that they're producing is not reaching the lysosome because it's unstable. When we give migalastat, which is an inhibitor, that enzyme becomes more stable and more of it can reach the lysosome. And when we measure the enzyme activity after they start treatment, we can actually see how it changes the activity. And in some cases, like this one, you can see how it normalizes. Now we're still early stages, and we still look monitoring these patients, but we can combine that pharmacogenomic with actually work in the lab to confirm whether they're actually responding as we expect them to. Patients are happy to go through this test. It's still normal blood test, so we're quite lucky to have those resources in Birmingham. Now, Gaucher disease, it's actually the same molecule, but it's a different enzyme. It's the beta-glucosidase, kind of down the line. So it causes deficiency on that enzyme that metabolizes the same molecule, produces a slightly different condition called Gaucher. And this one has different, again, multisystemic, but other organs are mainly involved. The liver, the spleen, the bones, the brain, the lungs, slightly different presentation, as I say. But it is a spectrum. There is not one with any condition. You have 3 different kind of conditions within one, and patients are moving within that spectrum. Luckily, it is also one of those that have several treatments available in the NHS, 2 enzyme replacements, which can give as an intravenous infusion, and 2 oral treatments, of which really miglustat is not that much use, but as you can see, the majority of them are either for Gaucher type 1 or 3, but there is no treatment really for Gaucher type 2. So there is a small cohort of the patients with Gaucher disease that don't have any available treatment. Now, eliglustat, I went into talk about this one because, again, approved by NICE 8 years ago now. Gosh, time flies. But we have to test these patients not for the actual gene variant that causes them to have Gaucher, but something called cytochrome 2D6, which is the enzyme in the liver that is going to clear out the eliglustat. So when we give it to patients, we need to test them to see how they're going to eliminate that drug. And if they are what we call intermediate or extensive metabolizers, that's the usual for 90% of the population, they have the normal dose, 84 milligrams twice a day. Some patients are what we call poor metabolizers, and they will only have they only need 1 capsule once a day because I'm not going to be excreting it very commonly. And part of the reason why I wanted to talk about this is because it's very relevant for the general population. This cytochrome 2D6 is the enzyme that metabolizes things like codeine into morphine. So if you are a poor metabolizer, when you take codeine, you're not going to get much pain relief. In some cases, the patients are called ultra rapid metabolizers. They have a lot very active 2D6, and they convert all of the coding into morphine, and they are at risk of having a overdose just with a normal small dose of codeine. This test, although not available in the NHS, is available for patients with Gaucher disease. So patients with Gaucher disease know the metabolizer status, but the general population, it is not a test that is a standard in the NHS, unfortunately. Just to give you an example, this is the kind of reports we get that will tell us the type of alleles they have, whether they are likely to be poor or intensive metabolizers, so we can determine what dose they should be having for elagolixotab when they start treatment. And then finally, I wanted to go back to mention about phenylketonuria, because there is also a treatment that will require pharmacogenomic testing before we prescribe it to patients or we offer it to patients. Because phenylalanine hydroxylase requires a cofactor called tetrahydrobiopterin. There are some patients with a specific gene variants that if we give them a lot of that cofactor, the enzyme can start begin to work, and therefore a company created a synthetic alternative called sapropterin, which took about over 10 years to get into the NHS because of cost issue. The company was asking for a too high price without giving too much evidence on how it would be benefiting the patients. But the idea is we overload the patient with that tetrahydrobiopterin, which is basically a water-soluble vitamin, and that will kick off the enzyme into converting phenylalanine hydroxylase into tyrosine. That's the originator name. Now we have a generic brand, and that's why it's available in the NHS, luckily. It had a very complex protocol. I tried to simplify it in a data poster for the Royal Pharmaceutical Society conference, trying to explain how we implemented this. So we have to first do genetic testing to determine the patient's variant of the PHH gene and see whether they're likely to respond or not. They have to go through a very strict dietary management all through their life. For the test, we have to make sure that they keep the phenylalanine blood levels within range and that we have maximized how much protein they can take. So because these patients have a very, very restricted diet. Some of them will only be able to take 3 grams of protein a day, which is less than a slice of bread. Once we are ready, we will give them sapropterin for a month, and then we will see whether, without any changes to the diet, so they have to stay on the same kind of diet, if we have a more than 30% drop in their Phe levels. Then, if that's the case, we move on to another extension phase where we will be trying to maximize how much protein they can take. So we increased their protein intake and keeping the phenylalanine levels within the recommended levels, which for an adult is less than 500 micromoles. Once they go through that extension period, then they can continue in it, providing that the Phe levels stay within the 75% of the time, stay within range. So I wanted to show you some of the results we got when we implemented it, and it is fascinating that even with the exact same gene variants, so I tried to plot it in a table, you will see that where the circle is, 3 different patients with exactly the same gene variants had a very different response to the treatment, from 40%, so that patient unfortunately couldn't continue after the first month, to 82% reduction in the Phe level in blood. That patient, which had a gene variant that we thought would be amenable and one that wasn't, didn't respond, actually got worse, the Phe levels. But that patient with a similar scenario, a variant that should respond and one that didn't, the same gene variant that will respond, had a 33% drop in the phenylalanine level. What we found out, and that was very reassuring, is that patients that were not going to respond didn't respond. We had to do 2 patients before we had the gene testing results back. When they came back, they hadn't responded, and it was obvious that they weren't going to respond. That has allowed us to manage the patient's expectations and trying to explain that if they're not going to respond, it's unlikely they will respond, and also to explain that when they are likely to respond, it's not always the case, and there are a few patients around that you will see that didn't respond on that table. But what it meant for the patients that responded was they had more freedom in their diet, and some patients, you could see that they increased the protein intake. The best one was over 450 times. So they were able to have close to a normal diet. And this is people that would grow up not being able to eat normal chocolate, not being able to, and never had an egg or protein, any kind of natural protein meat, and didn't know what chicken tastes like. So they were able to start introducing and managing a normal diet, which means being able to go out to a restaurant with your family and friends without having to worry beforehand what you're going to be able to eat, because in most cases for these patients, the only thing they could eat was a bowl of lettuce or maybe a small portion of chips, and that would have been everything in the menu available for them. So it did change significantly and improve the life of many patients. For some patients, it wasn't about increasing the protein intake. It was allowing them to maintain a better control of the phenylalanine levels. And that was important enough for them because it affects the mental capacity, and people and these patients, sometimes they will explain they have a brain fog when the phenylalanine level was high. So for them to be able to function normally, it was also just as important as, or even more than being able to eat more normally. And the difference, I mean, the diet make a big difference and transform patients like Sheila Jones that was never able to live independently, wasn't able to communicate or speak, and had to be in care all her life. We have patients nowadays that for the last 40 years that have been able to do their PhDs. They're very highly successful and high achievers. So but without the diet, they would have ended up same as Sheila Jones, unfortunately. But now, if we can, for some of them, and it's not the majority, unfortunately, those who have a minimum variance are more in the 20% of the PKU population, but at least they can start having closer to a normal life than they had before. So it has helped significantly. So just in summary, rare disease is rare, but I want to stress that rare diseases as a group, single group, are very common because 1 in 17 is very common. Just to highlight that remember everyone that the newborn screening program was started to diagnose and treat phenylketonuria, and that we are beginning to apply pharmacogenomics in rare disorders, giving some examples. It's not all about general conditions that we are already doing it, like cancer and some cardiac conditions. We are also little by little doing a little bit there. And that's me done. There's some references there at the back, and I don't know if there's time for questions, or haven't had a chance to look at the chat. But
thank you very much. That was absolutely so interesting and fascinating. And there is, if I go to the chats quickly, there is a question from Andrew Bell, who's asked that, thinking back to earlier talks on patient equity, does screening detect all variants, or is it skewed towards variants in the European population?
So the newborn screening test, it's a biochemical test. So that detects high levels of phenylalanine. So that is independent on gene variant. When it comes to do the looking for amenability for treatments, we are doing a whole genome sequencing, or we're doing exon sequencing. So we're looking at the specific spelling mistakes. So hopefully there shouldn't be any any discrimination or any patient miss because of uncommon variants. So we will be looking at what we've discovered with phenylketonuria, where and you will see in that table. Actually, if you leave it there, we did. Sorry, if you could go back that table, yeah. So the variants that the literature had confirmed that we're going to be responding, I did color them in green. The variants that the literature confirmed that weren't going to respond were the red ones, but there were a big number of variants, and probably that's a third or 40% of them that put on that example, that we didn't know whether they would respond or not. And obviously, any patient that we didn't know whether they were going to respond or not, we will allow them to go through the testing and see whether they were responding or not. As you can see, some of those variants actually turn out to be amenable to treatment. So we are conscious of the limitations of the technology. We try to be fair to patients and to give them the opportunity if there is any room for suspicion that they will respond. So it wasn't, but there is the risk when you test for one of these conditions that instead of doing whole genome sequencing, whole exome sequencing, that people are just going to look for variants that are more prevalent, and that would be a risk, because if you're testing for a particular variant prevalent in a population, patients who are not from that population will not be detected, and that's commonly happened, that patients from certain ethnic groups will be missed when you try for other groups. So that's tend to be the case when you do a pure classical biochemical analysis. If you look for homocystinuria, if you measure homocysteine, high homocysteine levels are going to be diagnostic. Then you go the genetic testing to see the spelling mistake or spelling variant. But when you do it the other way around, you're risking, if you go for prof testing, that those patients might be missed. So I think I'm trying to advocate for the biochemical labs to keep on doing the testing, and not to everybody think that genetic testing is the answer, because it's not always the case.
Yeah, definitely. That's about managing expectations, isn't it? But I just had a quick question, and it's kind of a general question, in that pharmacogenomics is offering this great potential in improving the management of inherited metabolic disorders. But thinking again of the development of new treatments, how can we ensure that new treatments, or what do you say about new treatments and therapies can reach patients faster?
Well, the NHS and the UK, in that sense, is in a very privileged position. We have different instruments to get them to patients sooner. NICE is becoming better at approving new treatments and new targeted treatments. So, and they don't require big numbers and long clinical trials. So the engine to approve and to fund those treatments is changing to adapt to that sense. We have in the UK something called early access to medicine schemes, with treatments that have promised scientific potential to help patients will be available if the company wants to make them available to the to patients before they have a license. They can go through this route, and they will become available in the NHS to the centers that want to participate in those programs, and patients can have access to treatment a couple of years before they have been available to the general patient population. So it's just kind of in between finishing a phase 3 trial and getting the funding approval, which a few years ago, when it was taking a couple of years, that meant some patients would have treatment 2 years in advance. And nowadays, luckily, the pace has increased, and some of these treatments are being approved very quickly. But still, the EAMS allow patients to get treatment a few months in advance. We have participated here in Queen Elizabeth in IMD in several, and a big advocate for them. I think they provide a lot of useful resources for patients, and try whenever I can to to encourage pharmaceutical companies to bring them over. Sorry, because I
Thank
think
you.
yeah.
Oh, sorry.
No. I just say that there are compassionate access programs, and we cannot get treatments, we will go through that route. We recently the NHS has individual funding requests, so if you can prove that your patient is the only one in the country that can benefit from that treatment, you can request it. It's virtually impossible, even when you have an ultra rare disorder. You don't necessarily gonna be able to prove that that's the only patient in the country. But what we found out was it might spark someone's curiosity. And when we submitted one for a condition called Tangier disease, which there's only 200 people in the world with that condition, we had one patient with it. The NHS England got curious about it, and instead of giving us the money for the treatment, they gave us the money to do an N-of-1 clinical trial to prove that the treatment was working. So even though, and then with the information to produce a clinical commissioning policy, so any other patient with Tangier disease in the future could benefit from that treatment. We're still working through the paperwork of the commissioning policy. It's a long process, but it is a tool that the NHS has available, even for ultra rare disorders. That Tangier is about 200 patients in the world. We have 2 patients with that condition. I think there's only another one in the UK. So that's even with those numbers, sometimes it's impossible to prove that you have a unique patient in the UK.
Thank you so much. And again, apologies for keeping you longer than we should have. Before I hand over back to Vicky, I just wanted to say on behalf of myself anyway, to thank you to everybody for really being, and to our speakers that have been amazing speakers and presentations. But thank you all for being part of this really important conversation to support patients and to support rare disease. It's all about action, advocacy, and advancing care. So thank you all to everybody. And I'm going to hand over now to Vicky to finish off.
Yeah, absolutely. I'm not going to keep people any longer on a Friday. Antonio, thank you so much for taking the last shift. I really enjoyed your talk as usual. You're so passionate about what you do, and it's lovely to kind of end on a note where some patients are actually kind of benefiting from the diagnosis with a treatment. And yeah, very excited as to where kind of pharmacogenomics will take us in the future. And to everybody that has joined us today, really grateful for you sort of coming along and hearing what we've got to say and what everyone's got to say. And if you could please help us out by completing this short survey. I've also put the link in the chat if it's easy to just copy and paste it rather than use the QR code. It's just really valuable for us to plan future events. And we will share the slides, and I'll share that link as well, just in case you don't have time to do it now. And just a very quick plug, it's very soon. We've got another